significant milestone in the history of higher education. We commemorate 145 years since the founding of the University of Medicine and Pharmacy, Iași. This institution has not only been a beacon of knowledge and research, but also a proud symbol of innovation and dedication to the health sciences. Over the years, UMF Iași has played a crucial role in shaping generations of doctors, researchers, and healthcare professionals who have contributed immensely to society. Thank you for being part of this celebration. This year marks another special moment as we celebrate 50 years since George Emil Palade, one of the most distinguished scientific figures born in Iași, received the Nobel Prize in Physiology or Medicine. His groundbreaking contributions to the field of cell biology earned him this prestigious recognition. In 1994, the Grigore Topopa University of Medicine and Pharmacy, Iași, honored him with the title of Doctor Honoris Causa, acknowledging not only his extraordinary achievements, but also his deep connection to his hometown. Ladies and gentlemen, it is my honor to introduce to you the esteemed director of the Grigore Topopa University of Medicine and Pharmacy, Iași. Under his guidance, UMF Iași has continued to thrive, building upon a rich tradition of excellence in medical education. Please join me in welcoming the rector to officially open this momentous ceremony, Professor Viorel Scripcariu.
Ladies and gentlemen, distinguished guests, dear colleagues, dear students, today we are gathered for a truly special occasion, a celebration of science, history, and inspiration. We are here to honor the life and legacy of an extraordinary figure in global science, George Emil Palade, as we mark 50 years since he received the Nobel Prize in Physiology. This moment calls us to reflect on the values that shaped his remarkable journey: curiosity, dedication, and a relentless pursuit of knowledge. George Emil Palade is more than a Nobel laureate. He is a symbol of excellence, a source of pride for Romania, and an inspiration for the global scientific community. His groundbreaking discoveries in cell biology have transformed the way we understand life at its most fundamental level, laying the foundation for many advances in medicine and research. His story inspires not only researchers and professors, but also every student aspiring to make a meaningful impact. During this opening ceremony, we are honored to welcome another remarkable figure of contemporary science, Professor Martin Lee Chalfie, Nobel laureate in chemistry in 2008. Professor Chalfie was awarded the Nobel Prize in Chemistry alongside Osamu Shimomura and Roger E. Tsien for the discovery and development of the green fluorescent protein, a tool that has revolutionized biological imaging. His presence is an extraordinary opportunity for our academic community to engage directly with a world-class personality whose work exemplifies the dedication, passion, and innovation that drive scientific progress. Another distinguished guest is Professor Ole Holger Petersen, a pioneering physiologist whose work has greatly advanced our understanding of epithelial cell ion channels, particularly calcium-activated ion channels, crucial for fluid secretion. Joining us online is academician and Professor Maia Simionescu, Director of the Institute of Cellular Biology and Pathology, Nicolae Simionescu in Bucharest. Academician Simeonescu spent 10 years training in Professor Palade's laboratory at Rockefeller University and Yale University in the United States. Her participation highlights the lasting impact of George Emil Palade's mentorship and scientific contributions, offering us a unique opportunity to benefit from her invaluable insights and expertise. We are so honored by the presence of 2 alumni of our university, also who have been awarded with Dr. Honoris Causa, Professor Daniel Gabriel Duda and Professor Cristiana Petri, distinguished alumni and Dr. Honoris Causa of our university. Another special guest is Professor Hegi, the director of the Center for Translational Medicine at Semmelweis University and close friend of our institution and of myself. Professor Andras Varo and Zoltan Konye from the University of Szeged also are our guests. We have a very good relation with Semmelweis University, and now through the help of Professor Hegi with Academia Europaea from Budapest. But this symposium is not just about the past, it's about the future. It is a bridge connecting the achievements of those who came before us with the aspiration of those who will carry the torch of discovery forward. It reminds us as a university community of our shared mission to foster a culture of excellence, to encourage bold questions, and to support the dreams of young researchers who will shape tomorrow's world. George Emil Palade's life story is one of connection and dialogue. Born in Iași, educated in Romania, but acclaimed on the world stage through the recommendation of his mentor, Grigore Tepopa, his journey reflects a beautiful balance of staying true to one's root while embracing the broader world of ideas and collaboration. We are privileged to organize this event here at UMF Iași, a university deeply committed to those very ideals. In 1994, George Emil Palade visited our university, receiving the title of Doctor Honoris Causa of UMF Iași. The recently inaugurated photo documentary exhibition and the commemorative volume launched yesterday stand as tributes not only to an exceptional scientist, but also to a man who carried his love for Romanian culture and values wherever his career took him. As we embark on this symposium, I encourage you, whether you are a seasoned academic, a researcher, or a student just beginning your journey, to draw inspiration from George Emil Palade's life. His passion for discovery, his rigorous approach to research, and his courage to ask the big questions are lessons that resonate across generations. At UMF Iași, we are proud to carry forward his legacy, shaping new generation of professionals and scholars who will build on the foundations he laid. Your presence today is a testament to the power of collaboration, curiosity, and shared purpose. With that, I am delighted to declare the symposium, the 5th anniversary of George Emil Palade receipt of the Nobel Prize in Physiology, officially open. Thank you for being part of our celebration.
Thank you. Allow me to introduce to you Academician Maia Simeonescu, President of the Biological Sciences section of the Romanian Academy, where she will also participate in this tribute session online and will speak about the legacy of the great scientist George Emil Palade.
Thank you very much for the introduction. Dear Professor Viorel Scripcariu, Rector of the University, dear Professor, researchers, residents, students, ladies and gentlemen, honored participants to this symposium celebrating George Palade legacy. I would like to extend a very special warm welcome to Professor Martin Lee Chalfie from Columbia University, Nobel Prize laureate in chemistry in 2008. We are very happy to have you here. Professor Chalfie is responsible for the discovery of the green fluorescent protein, GFP, that all of us are using today. Also, I would like to extend a warm welcome to Professor Ole Peterson, a well-known physician and physiologist, currently professor at Cardiff University in UK. We know, Professor, that you are the very first in the world to use the patch clamp technique on epithelial cells that is used by many researchers in Romania as well. I envy you positively for the Palade Award. Very warm greetings to Professor Duda, to Professor Apetri, and to all guests from abroad or from Romania. I'm really sorry I could not come to the beautiful city of Iași to admire the research results of your famous university. I am very attached to the university, to your university. When I returned home after 10 years in the United States, your university honored me with the title of Doctor Honoris Causa, and that moved and pleased me tremendously. But I am glad to participate to this symposium. I think it was an excellent idea. In the right place, George Palade was born in Iași, right time, 50 years since the Nobel Prize, and the right community, the community of the great experimentalist Grete Popa, one of the professors of George Palade. But let me tell you a few words about the man we are celebrating today. So the news about the Nobel Prize in Physiology or Medicine in 1974, it was awarded to Albert Claude, Christian de Duve, and George Emil Palade for discoveries regarding the structural and functional organization of the cell. You can see the picture of the three laureates. They were not together at the time in 1974, but they worked for a long time, and I would like to tell you a few episodes of their discoveries. The discoveries laid the foundation on which biology, medicine, physiology, virology, microbiology, and modern pathology developed. But I think it was more than that. Of course, this was very important, but it was a victory of human knowledge and human intelligence. But let's look a little bit at the journey of knowledge, I call it, the life of Professor George Palade. I would like to present you a few words about the stages of the discoveries, the journey of knowledge, and just few words. I don't have to tell you. You know very well why are his discoveries very important. So let's follow the course of life, which led George Palade from Hajdeu High School in Buzău, in Romania, to the Nobel Prize in Stockholm. So I was thinking that I will refer to 10 episodes, important episodes of his life. Episode 1. So I will present you the 10 episodes. Episode 1 was the family, the school, the education. George was born on 19th of November 1912 in Iași. The father, professor of philosophy, Emil Palade. Mother, a teacher, Constanța Palade. He had two sisters, Cim and Adriana Palade. What is important is the family environment was extremely important for his education. He got a respect for books and scholars and whatever it was knowledge around him at that time. So he went to the school at the Hajdeu High School in Buzău. He finished. He was an excellent student, excellent student, and but the excellency was even more clear in the years of the university years. So episode number 2 was student years. In 1930, he enters the UMF Bucharest, in University of Medicine and Pharmacy in Bucharest. There he had fantastic, excellent professors. I will remind you about Francis Greiner, the great anatomist, André Boivin, a fantastic biochemist, and Grete Popa, the most important and famous experimentalist of that time. So you can imagine the influence of these three people in his way of thinking and in his development or in his life. He worked in the Department of Anatomy. Interesting, he chose to have a very atypical doctoral thesis, the microscopic anatomy of the dolphin nephron. That shows more than that. That shows the curiosity which had this young George to understand how the structure of this kidney of the dolphin, which was like a grape, how the structure was adapted to the mammal marine life. So the functional adaptation of a mammal to marine life. It shows more than that. It shows it was a premonitory choice. It shows a curiosity of this young George and the curiosity and the application for the research. So he went to the faculty, he went to the army at the University of Medicine. He was associate professor at the University UMF from Bucharest. You can see here George Palade with his very good friend and colleague at the university, Professor Ion Juvara. They were friends for life. They started in the university and they were friends for life. Episode number 3, 1946. 1946. You know what happened in these years. So George Palade left Romania and he went to New York, to the United States, namely to New York University. There, something very interesting happened. He met Albert Claude, who came at the New York University to give a lecture. Albert Claude was working at Rockefeller University, gave a seminar with his recent results, on a new technique, a new technology which he introduced in science, namely differentiated centrifugation. So he was thinking, Albert Claude was thinking to use differentiated centrifugation of cells to isolate organelles and to study the structure at the electron microscope level. The electron microscope was introduced recently in the United States. George Palade was fascinated, but he had a fantastic intuition. He said it's very important and this is where he should be. So he moved to Rockefeller University. At Rockefeller University, where he stayed for 27 years, he started to work with Albert Claude, his porter, and what they wanted, to open the black box, which was a cell. Very little was known about the cell because the optical microscope attained his limits. So how to open this black box, the cell? So the idea was to make like a cell implosion, to disassemble the cell into components, and then to study the components, each component, at the biochemical and structural functional level. So the idea was fantastic. The concept was excellent, but was a big problem, and it was a big impasse. No success. The fractionation did not resemble those observed in the cell with a light microscope. So it's a special episode, number 4, when these first problems arrived at the big impasse in the study. What was the solution? So George was thinking that it was important to use another separation medium for the cell organelles. So that was the first saving idea, and it's amazing how he got this idea. He was inspired, he says, he was inspired by the culinary practice of my mother and aunt. So when they making the jam, they use whole fruits, and to keep them in a good state, they put the mother and the aunt put the fruits in a concentrated sugar solution, which we know we are all using this. So George had a great idea. He proposes to put the cell fraction in a concentrated sucrose solution. That was the first success, a tremendous change in the study of the cells. The second problem was a poor resolution of tissue at the EM level. The fixed tissue which you know we fix in glutaraldehyde and osmium, does not look like in microscopy. So the second saving idea was to adjust the acid fixative, osmium acid fixative, which was about 5.5 to pH 7, to neutral pH close to the lytic cell. That was the second success. So these two great ideas of George led to the landscape of the cell has become clear, and this is how the black box was opened. And this was the beginning of the discovery of subcellular structures. So episode 5, 1952, 54, and 56, time of discovery. They discovered that the cell organelles. George described very clearly and very profound the mitochondria, and later on the role as an energy source of the cell. He discovered the ribosome and endoplasmic reticulum together with Keith Porter, and the role of this, later on, of course, the role of this organelle and the synthesis of proteins. He describes the Golgi complex and the vesicles of the Golgi complex. So all this, I remember very well that he said every time he opened the microscope, he was discovering a new organelle. The plasma lemmal vesicles in endothelial cells, Emil Palade body, and the cardiac arterial granules, and so on. So you can see on the right side, that this complex structure was disassembled in organelles in components. The continuation of the period of discovery, with Sandy Palade discovered the synaptic vesicles of the neurons, and that was extremely important for the science of neurobiology. With Keith Porter, as I said, endoplasmic reticulum. But what is important that he discovers this ribosome. You can see on the left side here, these small particles, small osmiophilic black particles, which are the ribosomes. They were called Palade's corpuscle at the beginning, but he didn't like, he preferred to call them ribosomes. So then he describes the junctions with Merlin Farquhar and so on. So he was not content with this. After all these discoveries, as you can imagine, in the next episode, he's 40 years old, and he was asked to collaborate in very many other projects. Everybody in the States wanted to collaborate with him, but he didn't want to. He was obsessed. He wanted to answer his own questions. The role of these small black particles, ribosomes, and the role of if they are participating in the synthesis of new proteins in the cell. So he worked like a detective. He marked an amino acid to trace the place and the route and the formation of the protein. He had this vision that at the level of these ribosomes, a very important, a vital process for the life of the cells occur. The little bricks, amino acids, give rise to proteins for the whole cell. So he designed experiments. He found a biochemist, a very good biochemist, Phil Sikiewicz, And they started in 54 a very complex approach of structure, biochemistry, and function. This shows the way he was educated and his mind was at this complex approach to science. He demonstrates the chemistry of ribosomes, Palade's corpus with ribonucleic acid particles, and the formation was new proteins with which the life could not exist occur in the ribosomes attached to the ERs. And you have here an image of the cell with endoplasmic reticulum, ribosomes, Golgi, and the formation of proteins. For all this, in 1970, no, before that, in 1973, he moved to Yale University. He wanted to have more contact with the medical students. And in 1974, for all their discoveries, Albert Claude, Christian de Duve, and Georges Palade got the Nobel Prize for Physiology and Medicine. Now, what is interesting, you can see these laureates here. What is interesting that this episode 7, did not change Georges Palade's life at all. The questions in the next episode, which I marked here as episode 8, the question continued with the same flawless logic. What was the path of newly formed proteins? That was his question. The path of newly formed proteins. He has done original experiments, as I told you, radioactively labeling on amino acid, and traces the new formation of protein in time and route in the compartments in the intermediate station until the proteins reach the final destination. That was a discovery of the vectorial transport through the cell. It was a great discovery which was valid today. Then the question, how do proteins known to reach the target address? From a single source, proteins are sorted to different destinations. So the next question, how are the cell membrane produced? How molecules are transported from blood to tissue? This is a topic in which we were lucky to work with Georges Palade, transport of molecules from blood to tissue. These are projects on which he worked for 30 years with many very good, excellent collaborators, and which are being worked on today. I will go very short through this. I just want to say that we were privileged in 1969, Palade to invite Professor Nicolae Simeonescu and myself to Rockefeller University. By chance? No. Palade has not done anything just by chance. He was studying, I think, the problem that to invite us. He chooses. So we started a very good collaboration from 1970 to 79, and we continue this collaboration for another 4 or 5 years. So altogether, we collaborated for 15 years at least. We discovered together the processes involved in transendothelial transport, the role of vesicles, plasma-lemmal vesicles. We discovered transendothelial channels, specialized membrane, the existence of specialized membrane microdomains, the junctional differentiation along the vasculature, and many others. I want to say just what Georges Palade said about our experiment to build an institute in Romania. The Simeonescu experiment is worth trying. He calls this Simeonescu's experiment. Among experiments discovered in the selection microphotographs and writing manuscripts, Nicolae and Maya started working on the plans of an ambitious project, a new laboratory to be built in Bucharest. The initiative was entirely theirs. My role in this new project was to find together with them the ways and mechanisms to ensure the viability of this institute in Romania. And he really helped us a lot. He was the interface between us and the authorities and explaining them how important it is to have this Institute of Cell Biology in Romania. So he was a very good help. So in 79, we came back home from the States and we inaugurated the Institute of Cellular Biology and Pathology. What we wanted is to transplant in Romania the Rockefeller style of research for young researchers, for young Romanian researchers, which are just as clever as any other researchers. To introduce cell biology field in Romania and to start cell pathology and cell therapy projects. Coming back to George Palade, episode 9, 1990, he was 77 years old. He moved to the University of California in San Diego. He became Dean of Science. He still had NIH grants to continue his research, and he continued this. But also, in the same time, he was a Dean of Science of the university and brings a lot of commitment to academic and scientific excellence. He created a new department of cellular molecular medicine. Together with Marilyn Farquhar, they had a fantastic program, one of the best in the country. In 2002, he was 90 years old, he closed the laboratory. The university, which you can see here on the right side, the university building bears his name, George Palade. I think I will not keep you too long to listen to why Professor Palade's findings are important. What I can tell you except only that George Palade explains this in his Nobel speech. Cell biology makes a century-old dream possible. Analysis of diseases at the cellular level, the first step to control, cure the diseases of the century. And you know very well that the mechanisms discovered by cell biology were used later on by all the researchers in the world and in Romania in cell pathology. So apoptosis, which was discovered, is very well applied to cancer cells because we want that to kill these cells. The specific membrane proteins, which we all discovered in the cell membrane, are used now as drug targeting, the development of nanomedicine, or the autophagy in atherosclerosis. Episode 10. It comes a time. I call it departure, not separation. One day, October 7, 2008, at 12:01 p.m., George Palade left our world. In Del Mar, California, he was 95 years old. The professor wished the ashes to be scattered in Rocky Mountains in the States and the Carpathian Mountains in Romania. So the family, his daughter Georgia, son Philip, son George, Professor Radu Palade, Nimotache Zeletin, myself was accepted as a daughter or scientific daughter of George Palade. We went to the Carpathian Mountains in September 2010 at Vârful Cudor, and his ashes were scattered through the Vârful Cudor to the mountain. I call this George Palade came back home. But I want you to have at the end this idea. George Palade was a mentor, a master, and a teacher. He had 80 collaborators, published about 200 to 150 papers in 50 years of activity. So not too many, but they were all original papers. Each paper was a discovery or an interpretation of a discovery. His collaborators become leaders in the field in France, Israel, Italy, Japan, and Romania. His collaborators trained the second, the third, and the fourth generation of cell biologists. This is because Palade, then we all know that there is an obligation to the community to receive and to give. Palade was a man of all seasons. Look at him. He was a lover of universal and Romanian history, art, classical music, lover. He was a gourmet. He liked swimming, sports. He had a fantastic gift of storytelling and unaltered Romanian language. You can see him in different posters where we went together just a few days after each cell biology meeting to enjoy a few days. Or in the right side, you can see him on Thanksgiving. So George Palade I call the man who changed the realm of science. He's a legend. He's a legend that we like to remember permanently. A scholar with unparalleled logic, demanding of himself and those around him, a champion of distance, a seeker and a finder, a seeker and a finder. George Palade was a chance for science, a chance for every place he was in Romania, in the States, everywhere. He was really a chance to be with him and to be his collaborator. He's an illustrious explorer, followed by an army of other explorers, the collaborators. But the collaborators, as I said, became leaders in the field, and this is how cell biology was spread around the world. George Palade, a priceless legacy. The discoveries have influenced all the life sciences. Today, we pay tribute to Professor George Palade, and I admire you for making this symposium and this celebration in the UMF Iași. It is a celebration of the human spirit. His existence has inspired and inspires numerous researchers to seek the known hidden in the unknown, the truth from the untruth, the science from pseudoscience. Thank you very much for your attention.
Thank you so much for a true journey in George Emil Palade's life. Thank you again. And we continue. It is a great pleasure for me to introduce to you Professor Valentina Uivaroshi, Prorector of Bachelor's and Master's University Education at UMF Carol Davila, Bucharest, with a few words on this great day here. Thank you.
Distinguished Professor Viorel Scripcariu, Rector of Grigore Popa University of Medicine and Pharmacy, distinguished members of the academic community, honored audience, I am honored to represent the Carol Davila University of Medicine and Pharmacy to this symposium dedicated to George Emil Palade, probably the most outstanding graduate of the medical school in Bucharest. On the occasion of the University Days of Grigore Popa, University of Medicine and Pharmacy in Iași, I have the great honor and pleasure to convey on behalf of our management the warmest wishes for success and prosperity. On the occasion of the symposium celebrating the 50th anniversary of the Nobel Prize in Physiology or Medicine awarded to the Romanian-American scientist George Emil Palade, I bring a message from the rector of our university. I shall continue by reading the message addressed by Professor Dr. Viorel Ginga, rector of the University of Medicine and Pharmacy Carol Davila in Bucharest. To evoke the personality of George Emil Palade is a source of pride for the Carol Davila University of Medicine and Pharmacy, but also an opportunity to reiterate the fundamental values that mark the life and career of this Romanian origin scientist and to emphasize the legacy of his prodigious activity. As rector of the Carol Davila University of Medicine and Pharmacy, I am particularly proud to point out the contribution that the medical school in Bucharest had in the professional training of the illustrious scientist. George Emil Palade graduated his medical studies in 1940, got the title of Doctor in Science, and worked for a period of time as a university assistant at the Faculty of Medicine in Bucharest. In his autobiography, published on the Nobel Prize page, George Emil Palade mentions two personalities from the medical school in Bucharest that cultivated his interest in the fundamental biomedical sciences. Professor Francisc Reiner and Professor André Boivin, who taught anatomy and biochemistry respectively. An important role in George Emil Palade's career was played by Professor Grigore Popa, Francisc Reiner's successor at the department, who advised him to develop his exceptional qualities in a more performing research environment. Thus, in 1946, George Emil Palade decided to leave for the United States of America, where, after a short period spent in the biology laboratory of Dr. Robert Chambers at New York University, he obtained a research fellowship at the Rockefeller Institute in New York. The activity and collaboration carried out in one of the most prestigious scientific research centers in the world opened his way for the revolutionary research that culminated in winning of the Nobel Prize in Physiology or Medicine in 1974. The life of scholar George Emil Palade is a plea for the crucial role that values such as solid education, excellence, rigor, dedication, passion, teamwork, modesty, integrity have in building a remarkable career. Duty to the homeland and gratitude are other defining traits of George Emil Palade. Between 1942 and 1945, during World War II, Palade served in the Medical Corps or the Romanian Army. While he worked abroad, he sought to support Romanian research by inviting young researchers to the United States of America to specialize and then make a significant contribution to the organization and development of research in the country. Remarkable examples in this regard are Maia Simionescu and Nicolae Simionescu, academicians now, founders of the Institute of Cellular Biology and Pathology in Bucharest, and also of cell biology in Romania. Last but not least, George Emil Palade remained deeply attached to the faculty that formed him. Respecting the testamentary wish of George Emil Palade, his descendants donated to our university all the distinctions bestowed on him in over 60 years of prodigious career. These can be viewed every day by students and teachers, inspire them to persevere on the path of medical scientific research. In this regard, on the 110th anniversary of the birth of Professor George Emil Palade, the Carol Davila University of Medicine and Pharmacy opened a permanent exhibition bearing the name of the illustrious scientist. The main exhibits are the following: 15 national and international awards, the respective documents being signed by all the renowned personalities and heads of states, 49 national and international diplomas, documents signed by all the renowned personality and institution, 45 medals, some of which made of precious metals, such gold or silver, 3 decorations in the rank of Colan, 1 award with the golden statuette Victory of Samothrace, various documents that belonged to the Nobel Prize laureate George Emil Palade. George Emil Palade's legacy is impressive not only for cell biology, but for the entire scientific community. His work laid the foundation for many subsequent studies and research in the field of cell biology, and had a significant impact of the development of new therapies and medical treatments. He also influenced an entire generation of researchers and inspired numerous young people to follow the path of science. For all these reasons, bringing to attention the personality and legacy of Professor George Emil Palade is not only an occasion for great emotion and joy, but also a duty that you have toward the young generation, and I warmly welcome the organization of the symposium dedicated to the celebration of 50 years since the Nobel Prize for Physiology of Medicine was awarded to this illustrious scientist of Romanian origin. Respectfully, Professor Viorel Jinga, Rector of Carol Davila University of Medicine and Pharmacy. Thank you.
So, already all speakers greeted Professor Dr. Martin Lee Chalfie, showing respect and admiration for his work. I remind you that Dr. Chalfie is a neurobiologist known for his outstanding contributions to molecular biology and for developing the use of green fluorescent protein, GFP, as a genetic marker, a revolutionary technology for studying biological processes. Professor Martin Lee Chalfie is professor at Columbia University, where he has conducted groundbreaking research on gene expression and cell development. In 2004, he was elected a member of the National Academy of Sciences of the United States of America. In 2008, he was awarded the Nobel Prize in Chemistry jointly with Osamu Shimomura and Roger E. Tsien. He has published over 100 articles. His study, green fluorescent protein as a marker for gene expression, is among the 20 most cited papers in the field of molecular biology and genetics. Ladies and gentlemen, Professor Martin Chalfie will speak about the continuing need for useless knowledge. Thank you.
Actually, I'm not going to talk about that. Because I didn't realize what I was going to talk about until last night. But I've been thinking about this and thinking about George Palade and his career and his life. Before I really start, I want to thank the university here, people here. It is really an honor to be part of this celebration. So as you see, my title is Cell Biology Reborn. And I'll explain that in a moment, but we are here to honor, I don't have the control of this, do I? Let me see, how does that work? Whoops, let's go back, there we go. We're here, as you've seen, to honor the 50th anniversary of the giving of the Nobel Prize to all three of these individuals, but particularly to George Palade. I should say we're a little early. It will be exactly 50 years at 5 o'clock tomorrow, because the Nobel is always given at 4 p.m. in Sweden on the anniversary of Nobel's death, which is December the 10th. But we're close enough. And it really is to a remarkable set of discoveries. As it says here, for their discoveries concerning not just the structure, but the structural and functional organization of the cell. And George Palade's contributions to this, as we've already seen, are absolutely immense. So why did I call it this? Why did I call this talk Cell Biology Reborn? It's actually a quote from George Palade. At the Nobel ceremonies, or before the Nobel ceremonies, laureates are asked to participate, to contribute various things. They have to have a lecture, they have to give an autobiographical essay telling about themselves, and one person from the three is asked to give a speech at the banquet that is going to go to everyone. And in his speech to the banquet, all of this is on the nobelprize.org website, and it's wonderful to go back there and read it. But in his speech, he said the following. He said, "Cell biology, reborn, has created a new meeting ground in basic life sciences, for it showed that the unity of organization of living matter extends well beyond the level of molecules to the level of macromolecular assemblies, cell organs, and cells." That there was a university of cells at a profound level of organization of the parts of the cell. And when I read this, and as I've been thinking about him and his life, I did my postdoc with Sidney Brenner, who won his Nobel Prize in 2002. And Sidney is known for a rather interesting quote. And that quote is the following. Oops, no, let me go back. Before I talk about Sidney Brenner, I forgot about this slide. People like to say, and I've read this in several essays about George Palade, that he is the father of cell biology. Not really, because I think he's right by saying cell biology reborn. This man, E.B. Wilson, was a cell biologist at my university, Columbia University, and usually we say about him that he is the father of cell biology in the United States quite a long time ago. And he is because he wrote probably the most influential textbook about cell biology, and he was considered absolutely the most knowledgeable cell biologist, certainly in the United States, but also throughout the world. And his book, The Cell in Development and Heredity, is actually having its 100th anniversary next year. And the little reference at the bottom is of a paper that I helped write that really talks about the impact of this book. The American Society for Cell Biology gives a medal that's highest honor is called the E.B. Wilson Medal, and it's still going on to this day. But the reason I bring this up is because this book that's being honored is its third edition. And I think that's what it's really talking about is the rebirth of cell biology many, many times. So the first edition of the book he wrote in 1896. It was based on his lectures to university students. And it really defined the state of the field, which had really blossomed because of all the abilities that people had using vital stains, other types of stains of cells, and the light microscope. But it was restricted to that. But it beautifully describes the state of the field. 4 years later, he felt he had to completely rewrite the book because things had changed so much in just 4 years. And so in 1900, he wrote another one because he said, our concepts of cell division and fertilization have completely changed. I have to write the whole book all over again. And this book was around for a while, but then there was another really important change that led to the making of the book in 1925. And that, of course, was right after, as he was publishing the book in 1900, was the rediscovery of Mendel and the blossoming of genetics. And so by 1925, he felt that he had to incorporate all that had been discovered by genetics, what it meant about the chromosomes, the chromosome theory of inheritance and everything, completely changed the way, genetics completely changed the way people thought about cell biology. But I don't think he realized what amazing change was about to take place with the invention of the electron microscope. And that is where George Palade comes in. As you've heard, Albert Claude first introduced it, but it really is that George Palade just did astonishing work, as we've heard. His wife, Marilyn Farquhar, wrote this wonderful review about him. I've put the reference here. It is wonderful. That's where I got the picture from of him at the microscope. It's well worth looking at it. It's a beautifully written essay on his life and importance. It is very good. And so, but I want to point out, as I said, I worked with Sidney Brenner, and Sidney Brenner had a very wonderful line, which is this one. It said, progress in science depends often on new techniques, new discoveries, and new ideas, probably in that order. So he seemed to emphasize techniques. But by these criteria of what's important for the development of science, I would say that George Palade is a triple threat. He's all three of these things. So let me explain what I mean by this. He didn't just use the new techniques of electron microscopy and cell fractionation, which we've heard about. He improved them, and he improved them enormously. First by using osmium tetroxide fixation, and for cell fractionation, using sucrose buffers, which led eventually to sucrose gradients and the separation of organelles for biochemical analysis. Really, truly astonishing changes that enabled what's the next step? Not only new techniques, but new discoveries. And the discoveries were astonishing, because whereas the cell biologists before could see things staining differentially in cells, they really didn't know what these things were. They looked at the cells, there was something called the Golgi nets or Golgi apparatus. No one knew what it was. They looked at some other cells and they said, oh, this is a strange thing. We're going to call this ergastoplasm. What? No one knows what that word is anymore, but ergastoplasm. Anyway, he looked using his techniques at these cells and made amazing discoveries. So all of the pictures I'm going to show for the next couple of slides come from this amazing collection that's online at Yale of the various pictures he's taken. So here is a wonderful picture showing what a mitochondrion looks like and showing the cristae within the mitochondria. Just absolutely beautiful. Another discovery, the endoplasmic reticulum, seeing the ribosomes on these membranes, and just a fabulous structure. This was the ergastoplasm. This is what people didn't have any idea about. And he goes and he finds, and these are classic pictures of his work. Or here, the microsomes, realizing that the extraction of these components, the microsomes were first found by Albert Claude, but he didn't really know what they were until George Palade was able to fix them with Philip Sikovits and realize that they were small parts of the endoplasmic reticulum, the rough endoplasmic reticulum. He also defined the smooth endoplasmic reticulum as well in the cell. And then I think I have one more picture here. Yes. This is really a remarkable picture because you see on the left the rough endoplasmic reticulum, and on the right, all of the structures of the Golgi stack. And this is really an unbelievable picture because it begins to address a question of how do the parts of the cell work together? What are they doing? How do they interact? And of course, he was able to look at this, and so it's not just making the discoveries. He made an immense number of discoveries, but he was able to put them together and generate completely new ideas, particularly this one on how proteins are trafficked from the rough ER through the Golgi to the surface so that we get protein secretion and proteins put into vesicles and so on. And this is actually, you can't see it here, somehow it's been eliminated, but this is a picture taken from his Nobel Prize speech, also on the Nobel Prize website. And he was also somewhat renowned for being able to draw his own drawings, something I cannot do, but really quite wonderful work. So all three, techniques, discoveries, ideas, all of which he contributed. But I like this idea of the rebirth of cell biology. And what I want to end with is really the idea that it didn't stop with George Palade, that there have been a whole succession of new rebirths of cell biology as new techniques and new ideas have come into our understanding. So I'm going to list a couple of Nobel Prizes. One I'm going to be a little embarrassed about because it has my picture in it. But cell biology was reborn again with GFP. The ability to label cells or cell parts in living cells and be able to do a series of experiments. I'm going to take a little bit of time and show you a couple of pictures about GFP and what it's done. So the first one I'm going to show, these are two movies. GFP has been attached in the left-hand movie to a protein in the spindle that's going to be needed to divide the chromosomes at each nuclear division. This is 4 rounds of nuclear division in the fruit fly embryo. There's no cell division. And there you see the spindle forming and the division taking place. And a second time, and there'll be a third and a fourth. And the remarkable thing about seeing this in living tissue is that you're struck by how it's all happening at the same time. That they are, as Rosalind Silverman Gavrilo, who made this film and put it on the cell biology website, it is an example of something, she calls it in synchrony. So how does that synchronous activity take place? And that's led to lots of wonderful studies, and we do have a pretty good understanding of that. On the right-hand side, we have a different picture. This is of another embryo, but the same general time. But here GFP has been enters the nucleus because what's been attached to it is a small peptide called a nuclear localization signal. And if you have that peptide added onto anything, in this case GFP, it gets immediately sucked up into the nucleus. And so we're going to watch 4 rounds of division here as well, but now these are falsely colored. So the more of it in a place, it'll be closer to the red end of the rainbow. The less of it is going to be blue, and that's where the color comes from. But it's all from GFP. And let me start that movie. You see the nuclei break down. The GFP is all over the embryo. The nuclei start to reform, and they're sucking up the protein. And she names this after the Van Gogh painting Starry Night. It looks like that. And you can see here it's not synchronous. It's actually going from the lower right to the upper left-hand corner. We believe it's because her finger was a little heavy on the preparation in one part as opposed to another. But it shows you what you can see in a living tissue, which is really quite remarkable. Let me show you a couple of other things you can do with GFP. If you can put this into living organisms, and if you can see it, you can see things that you've never been able to see before. In our case, at the top are the 6 neurons that we happen to have spent maybe too much of my life looking at. But these nerve cells are very simple. They have, if you look at them, they have a very simple structure. Just above that white triangle in the top one to the left are the cell bodies. And you can see that there's a cell body and a process that goes towards your left. I don't think I have a Oh, do I have a pointer here? Yeah, here we go. You see this nucleus, and then the cell process goes here. That's the cell on the other side. It goes forward. All we have to do is treat these animals with a mutagen, wait a couple of generations, and look and see what the progeny are. And all of a sudden, we find a mutant that goes from a monopolar cell to a bipolar cell. And that immediately invites the question, how did that happen? How did this profound change in cell shape be caused by a simple mutation? And all sorts of other questions have been gotten. One can look for animals that don't have the cell in the wrong place, have too many cells, have the cells with too many processes, as here. And all of these lead to other questions about development and about cell biology. And finally, I want to talk about some very unexpected things, because all the work that George Palade did, and in fact that all of us do, we can never predict all the things that are going to happen, all the consequences that come out of that. I want to talk about two consequences where people have used GFP in ways that I'm still astonished by. So the first one was by Jeffrey Waldo, and he was interested in what was needed for proteins to fold. And he took a protein, he took the protein that didn't fold correctly in bacteria and attached GFP to it. Well, if the protein didn't fold, GFP wouldn't fold. If GFP wouldn't fold, you could shine blue light on it all the time, but you got nothing. And then he took his construct and he mutated the blue protein. It's not blue, but his protein, until he was able to change the protein so it folded correctly. Well, if it folded correctly, then GFP would fold correctly. And now, when blue light was shown on it, you would see that by green light. So he could just randomly mutate his protein and just look for bacteria that were green and pick them out, sequence the protein, and find out what actually made it fold correctly. I thought it was a brilliant experiment, brilliant idea. Another person is Lynn Regan, who was the chair of chemistry at Yale, and she had a really silly idea. She asked the question, what happens if you, what happens if you break GFP into two parts? And the answer is, it doesn't work. I'm sure everyone would have come up with that answer as well. But then she did something really brilliant. She said, well, if I take each half of GFP and I add to it a peptide such that the peptides come together, she used zinc finger proteins, then now they might be able to, or leucine zipper protein, they might be able to get that part together. What would that do to the two halves of GFP? And the answer was, they became fluorescent. She was able to reconstitute it. And so what she did in this was basically invent a way to look at proteins interacting with each other. Because if one protein had one part of GFP and the other protein had the other, if the two proteins did come together, the two halves of GFP would come together. And many people have done things subsequent to this, but again, a brilliant experiment. But it is cell biology reborn. Another example of that, 2014, the Nobel Prize to Eric Bedsig, Stefan Hell, and W.E. Morner was given to them because of the invention of super-resolution light microscopy, which gave another completely new dimension to things. And we're still looking at the consequences of this. And then my other example here is, so it's reborn again, and it's reborn another time with Jacques Dubochet, Yakima Frank, and Richard Henderson being awarded the Nobel Prize in 2017 for developing cryo-electron microscopy as another way of looking at proteins and protein aggregates and going even further than what George Palade was able to do. So we're constantly looking deeper into the cell. Cell biology is constantly being reborn. But I really like the idea of going back and reading about his work and his inventiveness and his scholarship and his enthusiasm, because I think that's one of the most wonderful legacies that a person can have. And so I'm really honored to be here to help celebrate this wonderful occasion. So thank you very much.
Thank you very much. Our next guest was the first in the world to use the patch clamp technique on epithelial cells. Professor Dr. Ole Holger Petersen, a Danish physician and physiologist, currently a professor at Cardiff University, UK, is here among us. Please.
Well, it's a great honor to be here today to participate in this very dignified celebration of George Palade, who was such a great figure. I'm a pancreatologist, and Peter Hegge, who will speak after me, is also a pancreatologist. So for us, George Palade is a real hero. As we heard from Maja Simonescu, Palade worked on a very large number of different tissues and had a very large kind of field that he explored. However, some of his most important work was unquestionably done on the pancreas, and in the Nobel Prize lecture, which was published in Science, as is usually the case the year after the award of the Nobel Prize, this Nobel Prize lecture was almost exclusively focused on the pancreatic work. So clearly he regarded himself, the work on the pancreas, as particularly important. So we already seen some of this. Martin showed another example of it. So as far as the pancreas is concerned, George focused on that for two reasons. First of all, because the pancreas is the great protein synthesizer in our body. There is no other organ that produces protein in that amount. It is really secreting proteins in bulk because we actually need a lot of pancreatic enzymes in our gut in order to digest the food that we eat. And the second reason was that even before he had, let's say, opened up the box and seen what is inside the cell, he realized that there has to be a movement in one direction in the pancreas. You have to move from the synthesis and you have to export the proteins at the other end of the cell through the apical membrane, and he realized that that is a big advantage in terms of understanding where different things happen in the cell. That still, today, is actually a huge advantage also in the cell physiology of the pancreas, and we have also explored that very much. The other element that he particularly focuses on in his Nobel Prize lecture is the process of exocytosis. And you see one of his famous pictures of exocytosis, the fusion of the secretory granule with the plasma membrane and the opening at the point of fusion between the interior of the granule and the lumen of the acinar cell. I will later come back to the fact how remarkable this is to have captured that. And for those who followed Palade's work, it was interesting that he basically for many years only had two pictures of exocytosis, and this is one of them, because it's actually damn difficult to catch it. It's very, very short-lasting, and I will show that in real time in a moment. In his Nobel Prize lecture, he also has a very nice acknowledgement, which I have put there on the top of this slide, where he pays tribute to his many outstanding collaborators. And I will particularly mention here Jim Jamieson, who did most of the work that actually resulted in the elucidation of the secretory pathway. And I will talk a bit more about another of his collaborators, George Scheele, and we'll come to that in a moment. Now, as a young and fairly unknown scientist, I was obviously extremely pleased when I saw George Palade's publication in Science, because he actually cited one of my papers in it. And so I thought that was pretty cool, as we say today. And he made a particular point about the stimulation of secretion. So although it was not his primary research subject, he was interested in the way secretion is switched on and off. He understood, of course, immediately that it would not be sensible for the pancreatic cell to continuously secrete, because it's an expensive process bioenergetically. It costs ATP to secrete. So he understood that in the kind of resting conditions when we don't eat and don't have to digest, there should not be secretion. It is not meaningful. But in the moment we take food into our system, then of course you need the enzymes to break down the food that we have consumed so that it can be broken down to small components that can be absorbed in the gut. And so he was interested in the finding that we published in 1973, that one of the, well, both the major stimulants of pancreatic secretion, namely the neurotransmitter acetylcholine released from vagal nerve endings and the circulating hormone cholecystokinin, actually depolarize the plasma membrane of the Eisner cell. That is, they reduce the electrical potential difference across the basolateral membrane. And it was mentioned, I think, both by Maria Simonescu and also by Martin Chalfie, that Palade was very keen on the principle that the same rules, the same mechanisms occurred again and again in the body, and that the body used the same tricks again and again. And so the reason that he was keen on this particular finding of ours, that there is a depolarization, is that he saw an analogy with what happens in the nervous system. And as you can see here to the right, in a few years before, Alan Hodgkin, who of course won a Nobel Prize in 1963, and his collaborators demonstrated directly the rise in the calcium concentration that follows trains of action potentials in the nerve. And so George liked the idea that obviously he understood there is a secretion process from the nerve endings, it is calcium mediated, and it is initiated by a membrane depolarization. So he felt when he saw that the pancreatic Eisner cell also depolarizes, that must be the way it works. So he saw that we have the same mechanism in the nerve ending and in the pancreas. This turned out not to be quite right, actually. But George was very keen on this idea and defended it quite strongly. Now, he could have cited another paper on mine that was published a little bit later that same year, and which in some respects is actually more important, but it did not fit in so well with George Palade's thoughts. And in my sabbatical at the University of Cambridge in 71 to 72, I met an American scientist, John Williams, and he did not work on the pancreas. He worked on the thyroid gland. I worked on the pancreas. And this paper that we published together, I should say actually that in those days the Journal of Physiology insisted on author names being in the alphabetical order, so there is no significance to the alphabetical order here. In fact, the first author, Keith Matthews, was away from Cambridge the whole year on sabbatical in Geneva, and both John and I were nominally working with him, but he was actually not there. So it was the work of myself and John. And we used radioactive calcium and demonstrated, as you can see here in the top, that stimulation has an immediate effect on calcium fluxes. It releases pre-labeled calcium from the gland, and that is then followed very quickly by an increase in secretion. We also looked at the idea that membrane depolarization might be the trigger for secretion. And actually, if you expose cells to high potassium concentration, which will depolarize the membrane, then you will get calcium flux and you will get secretion. However, if you block the muscarinic acetylcholine receptors with atropine, nothing happens. So we reasoned that it is actually the depolarization of the nerve endings in the pancreas that releases the transmitter and that causes the secretion. Now this is a little bit important for what follows because that's where a big disagreements occurred. So the general idea was actually formulated by Bill Douglas, who also spent most of his life at Yale University, namely that there would be an interaction with the membrane for a transmitter, there would be an increased membrane permeability, there would be entry of sodium and calcium, depolarization, and then secretion would occur. Now, of course, one looks for models of how calcium entry could occur, and a very good example is in the heart, where calcium entry is vitally important and is dependent on a very large depolarization. So you see the action potential here taken from this excellent review article by Andros Varro, who is of course with us here today. And you see that you get a fantastic change in potential from a resting potential of close to minus 90 millivolts, and not only is it depolarized, it actually reverses the potential and becomes positive. And this actually then allows the activation of voltage-gated sodium channels and voltage-gated calcium channels, and calcium enters the cell. And so this is what happens in the nerve cells and in the heart. However, when we looked at this in more details, it turned out that in the pancreas, it's actually the other way around. It is not the depolarization that causes the calcium signal, it is the calcium signal that causes the depolarization. And excellent, my very first PhD student, Noriyuki Iwatsuki, who was a surgeon, so with some credit to surgeons here, he came to me, had never done physiological experimental work, but he turned out to be a very skilled electrophysiologist. And he established this configuration that you see in the picture in the top right there, which was our kind of working constellation for very many years. It's still the era of sharp intracellular microelectrodes. And we discovered that all the Eisner cells in an Eisner unit are actually very strongly coupled via gap junction. It's like a one big cell. And so we can actually inject calcium in one cell, and we can record the change from the other cell. We can apply acetylcholine very close to the cell. And in this experiment illustrated at the bottom there, you see that the external application of acetylcholine is mimicked by the intracellular application of calcium. So by injecting calcium into the cell, we can completely reproduce the effect of acting by acetylcholine on the outside and our CCK. So that obviously changes the picture quite radically. Now, in more recent times, where we have been able, with regard to more advanced microscopy, to look at several cells and calcium signaling directly, we have been able to directly see the neuroglandular transmission, and we can again use high potassium to depolarize all the cells in the pancreas, but only two cells will show a calcium signal, namely a neuron and the Eisner cell. And at the bottom, you see the important experiment again, high K, depolarizing the cells, calcium signal in the neuron, followed by a calcium signal in the Eisner cell. But in the presence of atropine, you only see the calcium signal in the neuron, and it's gone in the Eisner cell. So that, in a way, seemed to settle the matter. So this is all work on the mouse pancreas. Now, many people dislike intensely any kind of discussion about species differences because it's very messy and very irritating, and George Palade also didn't like it much. He always worked on a guinea pig pancreas, and indeed, as you saw in one of Martin's slides, they were taken very specifically from the guinea pig pancreas. So George Schiele, one of Palade's close collaborators, followed up George's idea that depolarization of the pancreatic cell membrane is actually important for secretion. And he provided evidence that actually looked very convincing, and you see this in the slide to the right. So potassium depolarization causes secretion if there is calcium in the outside solution. So you see here with 2.5 millimolar calcium, a huge secretion, with small amounts of calcium, much less. And if there's no calcium on the outside, the potassium depolarization doesn't do anything. So he did accept our idea that there must be also some release of calcium from intracellular stores, as you see in the diagram from that JBC paper, but nevertheless suggested that the depolarization was critical and that there had to be translocation of calcium across the plasma membrane in order to stimulate secretion. So obviously this was a challenge for us, and we had to look into this. So we adapted the method of electrical field stimulation, where you isolate a piece of pancreas, put a couple of electrodes on the outside, and by inducing an electrical field, you actually set up action potentials in the nerve endings in the pancreatic tissue and release neurotransmitter. And it works very beautifully, as you can see. We measure amylase output with a very high resolution technique, fluorescent probe technique. And you see we actually, in a way, reproduced the experiment of George Schiele. Yes, he was right. Nerve stimulation will actually produce secretion even in the presence of atropine. So at that point we thought, okay, well, actually it does seem that they have a point. However, then we did one more experiment. We used the very specific nerve action potential blocker, tetrodotoxin, which is a very specific inhibitor of voltage-gated sodium channels. Very specific inhibitor. And you see the response is gone. And when I saw that, I immediately said yes. So what is happening here is we are stimulating nerve endings, and they are releasing in the guinea pig, in addition to acetylcholine, another transmitter. At that time, it was known that there is very often several transmitters released from one nerve endings, and that was the point that George Schiele had not appreciated. We looked at other things. We looked at calcium flux, and we again noticed that in the presence of atropine, although secretion happened with nerve stimulation, there was no change in calcium flux. Also, we looked at membrane potential changes. Again, in the presence of atropine, there was no membrane depolarization of the Eisner cell. So this very clearly led to the conclusion that actually the membrane depolarization does not elicit secretion except if the nerves were to do something else than just releasing acetylcholine. So it was known that in many tissues, acetylcholine from vagal nerve endings is released together with vasoactive intestinal polypeptide. So we thought we'll just try that. And indeed, it turned out that VIP, as you can see in the upper right corner there, vasoactive intestinal peptide, produced a change in cyclic AMP. Now, this is an interesting point. One of the early meetings I had with George Palade was at a meeting of the American Society of Cell Biology in Baltimore. And there was a big clash between Mickey Schramm, an excellent biochemist from Jerusalem, and George Palade. So Mickey Schramm had provided evidence that in one of the salivary glands, the parotid gland, it is actually not a calcium rise that initiates secretion, but a rise in cyclic AMP. George Palade hated this idea, and there was a big clash at that meeting between Mickey Schramm and George would not accept that, actually. He just said, no, something is wrong, actually, there, because it has to be calcium. It cannot be cyclic AMP. But actually, Mickey Schramm was right. There are some tissues where actually cyclic AMP is an initiator of secretion. It may well sensitize the secretory mechanism to calcium, but the initial response is actually cyclic AMP. And then you see here that everything high K, field stimulation, reproduces what vasoactive intestinal peptide can do. So we published then an additional paper also in JBC and had this model here that there actually are two secretory pathways in a guinea pig, and this cyclic AMP pathway does not exist in the mouse, which therefore for that reason is simpler, but it is actually a possibility, and in fact it is not a depolarization of the Eisner cells that causes secretion. Later on, we were able to demonstrate very directly the calcium release from the endoplasmic reticulum. You see here a maximum secretory response releasing calcium from the ER, and we can monitor the cytosolic calcium concentration by using an endogenous sensor in the cell membrane, namely the calcium-activated chloride channels. And the more physiological situation you see to the right, where every single calcium spike captured here by the recording in the patch clamp configuration of the calcium-dependent chloride current is associated with the release of calcium from the ER, and then in the interspike period, calcium is reaccumulated again. And of course, we now know fully how that works through the discovery of Mike Berridge of that acetylcholine. Just one example, interacting with receptors and via G-protein proteins, we break down phosphatidylinositol bisphosphate, generate the small water-soluble molecule IP3, which then can release calcium from endoplasmic reticulum. And this discovery was made also in pancreatic Eisner cells, another example of a fundamental discovery that was done with the help of the pancreas. And we can use, again, the patch clamp configuration to record the calcium spikes at high time resolution. And here again, we focus on the calcium-activated chloride channels. And we can see these calcium spikes in the complete absence of calcium in the external solution. Another big difference from the heart and from the nerve cells, which cannot secrete in the absence of external calcium. So, finally, to come back and also actually illustrate the correctness of the main finding and the main postulate of Palade that it is actually calcium that ultimately controls secretion, we were able to monitor that very directly. So finally in '93, in a paper in Cell, we were able to demonstrate that the physiologically relevant calcium spikes are occurring as short-lasting repetitive spikes, which occur only in the secretory granule part of the cell. And by another use of the patch clamp technique, where we can measure the capacitance of the membrane, we can actually look directly at the insertion of the secretory granule membrane into the plasma membrane. Because in a moment when the secretory granule membrane fuses with the cell, as we saw in that picture from George Palade's work, the membrane area is increased. But there is very quick recapture of that membrane, and that's the reason that it is so difficult to see the exocytotic event, as I mentioned. And here you see a single calcium spike resolved here, where we actually, in order to plot everything upwards, don't show the chloride current, but we show the chloride conductance. And you see here that the calcium signal actually starts a bit earlier than the exocytotic event, which makes sense. So we activate the chloride channels a bit earlier, which activates the fluid secretion, and that's very important because you need the fluid to wash out the enzymes of the system. And you see how short-lasting a single secretory event actually is. In many cases, as short as only a few hundred or milliseconds. And that's why it is so difficult to capture the exocytotic events, but George Palade managed skillfully to do exactly that. So here in real time, we see the exocytotic secretory event, and we can see how, as the calcium concentration rises, we insert more and more membrane, because in the pancreas it's very often a compound exocytosis. It's not just one granule that fuses, but other granules fuse with the fused granules. And then you see the recapture of the membrane. So in the fullness of time, this stimulus secretion coupling sequence was unraveled much more, and of course, as always, it turned out to be a great deal more complicated than originally thought. And these two main stimulants of secretion, namely acetylcholine and cholecystokinin, induce subtly different calcium signal patterns and also work via somewhat different intracellular messenger pathways. But I will not go into detail with that because that's not so important in the context of this talk. So the International Association of Pancreatology honored the memory of George Palade by creating the George Palade Medal, which is given each year to one. And the very first such medal was naturally given to Jim Jemison, who played such an important role in the development of the story as far as the pancreas is concerned. And I was also delighted that my close colleague in Cambridge, with whom I worked, John Williams, got this medal some years ago, and of course thrilled that I finally also obtained this. So I will conclude my talk by showing some of the collaborators. Many of them, unfortunately, I did not have time to talk about their work today. And to the right, I show many of the people who inspired me. Again, unfortunately, I did not have the time to talk about Peter Hege's very important work on the pancreatic duct system, but he will speak himself, of course. He is very much the future of pancreatology. We talked about the past, but now here with Peter Hege, we have an example of somebody who definitely is the main person of the future, and I'm pretty sure that the Palade Medal will come to him in due course. And I was delighted to hear that he will be made an honorary doctor at Bucharest next week, so we can already begin to celebrate the future. So I thank you for your attention. It was a privilege to speak here at this meeting about George Palade, who really was fundamental to the work that all of us who are pancreatologists are doing.
Thank you so much. We continue with Dr. Peter Hege, a member of the Hungarian Academy of Sciences and director of the Center for Translational Medicine and at Semmelweis University in Budapest. His groundbreaking discoveries in pancreatology have elucidated the critical role of ductal cells in pancreatitis. So thank you for being here. We will listen to you.
So, dear Mr. Rector, dear guests, students, and it's a really great pleasure to speak here today, but after two giants had a speech before me, it will be difficult to speak, but I hope I will give you some idea what I think about science, what I think about discoveries, what I think about the future of sciences. And I am a clinician. But a physician scientist doing science and seeing patients every day. And in clinics, we usually start with disclosure, but today I will have 3 disclosures. Let me start with the very first one. My first disclosure is I love science. And I will tell you very clearly why I love science with 2 showcases completely outside of pancreatology. One showcase is if you look at, for example, somebody is coming to the unit, 25-year-old, hallucination, delusion, social withdrawal, everything, has a schizophrenia. How to treat these patients? We have two choices. One of them, lobotomy, half brain take away, ice cold baths, physical restraints, or the second chance is to use antipsychotic drugs. Of course, if we choose today, it's not a question which one to choose, but I tell you that in 1871, this was the protocol of treating schizophrenia. But after, based on understanding several mechanisms which lead to understand the disease, we use the second treatment. But okay, Peter, you can say that this is far, far away, but not. If we look at another inside gastroenterology example, after abdominal pain, peptic ulcer, and so on. So one possible treatment is B-road resection. I think some of you in the room still remember when in a surgical department, 1/3 of the patients, actually the stomach were cut out. Why? Because we didn't understand the disease and there were no specific treatment. And the second option is to use proton pump inhibitors. But why we can use proton pump inhibitors? Because somebody discovered the hydrogen potassium ATPase inside the stomach. So this one was in 1975. There were no surgeons who grew up without doing B-road resection. Now a resident doctor even not seeing this disease. So if somebody asks me, what is my opinion in science? If no science, no present treatment, and no future. This is as it is, in my opinion. I was really, really very happy when I came here in February and with the rector we discussed the importance of science, and it was really a privilege for me to hear how much in Yash people love science, and they are dedicating actually this year to the 50-year anniversary of George Emil Palade. Really congratulations for Yash, because he had this fundamental discovery. I don't want to go into the details. We heard from Martin and Ole lots of fun, but concerning the pancreatic acinar cells, this was the very first visualization and the very first even that we understood that the pancreatic cells, how they secrete actually zymogen granules. It's for me, a pancreatic doctor physiologist, just very, very good to hear that he used guinea pig. Actually, guinea pig, the only animal which secrete the same amount of bicarbonate, 140 millimole like human. So for me, I always use guinea pig for mimicking the human situation. So, and this was the one when we were front, to be honest, and sorry about that. At that time, I didn't even know in February that George Emil Palade was in Yash, but when we met with your rector and stand there, I asked, why do you call this auditorium George Emil Palade? He said, because he was born here. So this is when I learned it, and I'm happy to come back. So actually, today, the talk is from science actually to society, but to get, we also need curiosity for young people and to bring so the career. And I just show you what, as I mentioned, I believe concerning science. So you need to discover things first, because if a healthcare challenge is coming, you need to find community solutions. What you need is, and I am so happy to see so many young people upstairs as well, that you need talented youth and creative ideas. But look at the danger today. This is a summary of in young secondary school students that the interest in science continuously decreasing. So we need to think about a new model of education, how to help young people, to involve in scientific activity. The other issue is, however, that we need healthcare professionals, doctors, psychologists, dietitians who are using this knowledge. We usually say you have to discover twice something. First, you need to discover what it is, and after you need to discover how to make it useful. So you need healthcare professionals. The question is, do we use today the scientific knowledge efficiently? The answer I will show in a Eurostat data, it shocked me first when I saw it. Eurostat make a data to show how many deaths today under 75 would have been avoided or delayed if we use all the scientific knowledge generated in the past. And here are the data suggesting that in Europe under 75, 1.7 million people dies. However, 1.2 is avoidable. And especially in Eastern Europe, like in Romania, Hungary, it's 80%. 4 of 5 deaths in this region, including Hungary, could have been treated or prevented if we use the knowledge. So therefore, we need doctors. We need society who are actually using this. And it's very clear that we have always to look at new approaches, and we need to look, new models, because if we use the same thinking how we created the problems, we will actually never sort it out. So therefore, in Hungary, with the National Academy of Scientists Education, I'm so happy that András Varró is here today, and also with Academy of Europea, we built up 2 programs, and this was the second one which actually let me meet the YASH people. One of them, how to involve new students, secondary school students in the program. And this is actually, we have now in Hungary over 3,000 secondary school students involved in secondary program, and the other is concerning the PhD program for healthcare professionals. We'll teach the healthcare professionals how to use the scientific knowledge. And luckily, actually, this is a new model with Academy of Europea and the National Academy of Scientists Education, and we were very happy that we could publish these 2 models. And why I am telling this? Because I am really very happy to be here. Because in Szeged, for these 2 programs exactly started almost the same we are here today. Because we celebrated the past. We celebrated the 75th anniversary of the Nobel Prize of Albert Szent-Györgyi, who was delivered his research there. Now we are here at the 50-year anniversary of George Emil Palade. I do hope, and I am sure, that in this town, the same project with the secondary school students and PhD students can go through. So what I think is needed, first we need brilliant ideas to discover new techniques, new things. After, we need to do translational science, basic science, and we need clinical science to bring it back. So let's start with the discovery science, and this is where George Emil Palade and his colleagues Albert and Christian received the Nobel Prize in 1947. But there are other models as well, and I'm so happy that Ole Petersen could come, Bert Sakman and Evry Neher received the Nobel Prize for discovering the patch clamp technique, and both of them always mentioning that Ole Petersen, he used it in exocrine cells, and also actually he was the very first one showing that if acetylcholine stimuli coming elevate the calcium signaling, this lead for the secretion, he also should have been among them. And after, it hasn't stopped, because we also need, markers for the cells. And I put these 3 techniques because what I will show you in translational science, I use these techniques to discover new mechanisms in pancreatology. So the focus during the discoveries wasn't on utility, but on understanding the underlying processes. It's extremely important. Nowadays, I hear from plenty of universities, no, basic science and governments are not highly important. Highly important is the knowledge we can use and for society benefits. If this discovery is not happening, none of my experiments, I will show you later, would have happened. So thank you very much, Martin. Thank you very much, Ole, because without your essentially, these experiments would never ever been performed. So here is my second disclosure, confess, I love pancreas. I can't hide it, and I will not, for sure. So, and I started reading the articles, and this is what I always suggest to my students, read all the articles, because there are plenty of discoveries then which was forgotten. And this was one of the paper I read, and I'm sure Ole met Henri Sarl, who was one of the biggest clinical pancreatologists in Marseille, and published an article, and he showed for the very first one that there are, if the alcohol-induced pancreatitis, there are some these mucus gels inside the pancreatic ducts. And he asked the question, why? And he looked at this phenomenon, and actually what he realized and put it down, that the chloride and sodium significantly were higher, those patients sweat who actually were alcoholic and had this mucus gel. However, at that time, there were only a single sentence in the discussion part which was written, we give these facts without drawing any conclusion about this complicated problem, because they saw it's an adult mucoviscidosis. And after what's happened in '89, the CFTR was discovered and published in Nature. So after that, they knew that this is actually a chloride channel. But because it was published 24 years later, earlier, but people are not came back, pancreatologists, to look at this. However, this is exactly showing a mechanism of alcohol-induced pancreatitis. And if we look at CFTR, this has been shown very elegantly in several models that the genetic mutations inside CFTR can lead to pancreatitis. This was clear with plenty of models. However, no one asked the question, what Henri Salle showed there, that if we go actually further and we look at how pancreatitis develop, whether only the genetic mutations of the CFTR will lead to pancreatitis, or this is a general mechanism, the toxic factors can also act via the CFTR. So our major question was that, because if yes, this could be a very first therapy in pancreatitis, which has no therapy today. So we did huge amount of experiments actually, and I was sorry that Palade didn't like cyclic EMP, because with ductal physiologists we love cyclic EMP, because both the chloride bicarbonate exchanger and the CFT are working through. But here is the technique all I used, and Bert Sackmann, they are discovered. We measured with the patch clamp the chloride exchange, and we could see that if we give ethanol and also fatty acids, it's just inhibiting the CFTR channel. And when we used the protein what Martin discovered, so you can feel that discovery science essential to do translational research. Actually, in the guinea pig, what also George Emil Palade used, we not used the acinar cells. We looked at the CFTR channels, and it was on the membrane, but when we used alcohol, it actually moved back, fold back to the translocated, actually to the cell. So we realized that CFTR is really a target of toxic factors, ethanol. So, and this is what I am saying, that I love working with students. I can learn as much as from the students than from my mentors. And I ask my students how we can test a theory in human, not in guinea pig, that during alcohol, the sweat chloride channel inhibiting and the chloride are going. So my students immediately answer the questions that we need to find volunteers. Who is happy to drink alcohol? So I sent an application for the ethical committee, and we got for 10 volunteers who are happy to drink actually 400 milliliters of vodka within 2 hours. And we measured the sweat chloride channel. So by the time I arrived back to the lab, from the 10 sheets, 9 were signed by my students. They wanted to be volunteers in the study, but they left out 1 glass of vodka for me. So I was the patient's number 10. And when we measured it, actually, this study, it didn't change at all. We reached 20 millimolar alcohol concentration, but didn't move anywhere or sweat chloride. My students came back. Peter, what did you say last time when your experiments failed? Repeat it. So they come back that we want to repeat this experiment. So I was thinking, oh my gosh, the grant committee will see about the compost I bought from the grant, vodka. I said, it's not possible, so let's find another way. And I don't know Yash, but around Szeged we have some professionals, if you go on the streets, so and we found some of them. And when we measured then chloride concentration, it was 80, 4 times higher than us. What shocked me actually, after 400 milliliters of vodka, and I reached 20 millimolar ethanol concentration, they reached 100 millimolar, which means they should have drink, 2 liters of vodka that I would have died. And one of the most difficult review ever I had in my life was that in gastroenterology they asked us to look at when these guys' alcohol concentration 0. So Szeged looked like that there was somebody like in the picture and the PhD students next to them, because my PhD students followed them to remeasure the sweat chloride concentration when the alcohol concentration dropped. But it was very clear that this is reversible. So if they stop drinking alcohol, CFTR can go back to the membrane and their activities actually come back. I don't want to go very much into the details, but I had really the privilege to publish this review in the Physiology, Biochemistry, and Pharmacology with Ole, who had really brilliant experiments actually on acinar cells. So after that, we also discussed the students, what should be the next experiments? Now we see that ATP decrease also inside the cell, mitochondria damage. So they said, okay, so let's try to give back the ATP if it's inside the Packato mechanism. And it was also Ole's lab with David Criddle showed that if you actually, inside the acinar cells, if you give ATP back to the cells, the toxic calcium signal became to be normal. We saw exactly the same in the CFTR channel. If you give ATP inside the cell actually, again, the CFTR activity start growing. So, and as many people in their life, I look at actually a closed road because I needed patience to continue these experiments because we had idea how to bring it there. So this was the time when I moved from the University of Pécs from Szeged there, and I must say that it's really a great pleasure to do something what people believe that you can't do. And this is the third part, and this is why I am saying that not only discovery and translational science is needed, that clinical science as well. So we looked at first the meta-analysis, which is a very nice form of the understanding, the current knowledge and the current biases, and it showed easily you can bring ATP to the cells not only via like a patch pipette, but if you use enteral feeding, actually with the tube feeding, you can bring energy back to the cells. So, and if they used energy feeding and not a nilperose diet, this was always beneficial. So innovation starts when the existing tools are not enough. So for that, we needed to build up a clinic. And in the clinic, there were 2 very important units. One unit is a multidisciplinary with lawyers, biostatisticians, IT coordinators who led the clinical study, and we needed to build up a room. I am so happy that in Budapest, our 30 beds pancreatic clinic, we have now 38 PhD students. So we have currently more students than beds. That's really brilliant. So using this model, this is what we started to build up. We started looking at what's happening if we bring the energy back to the cells. So we use a nasogastric tube feeding, and it seemed from this cohort that the patients spend less time in their hospitals, and actually the amount of severe and moderately severe pancreatitis is less. However, in clinic, there are also methodologies like PCR, Western blot, and all the others, because the final confirmation are the Western blot, the randomized clinical trials. And also my students, I learn a lot. They are really very creative. So I told them, let's do a study and let's find an acronym for the study. And a couple of weeks before, the Spanish did a water study. And because they are Spanish, they checked saline and ringer lactate. They called the study Sangria. This was really a very nice idea. So I told them, find a name, which is like everybody knows, that this is actually Hungarian concerning the general utilization of energy, and they called the study the Gulyás study, and we published the protocol, and it was, we just finished the study and made the analysis. And this is another message. I remember in the 90s when we started using clinic, everything which is logical for basic science. So this was a very logical setup, and we strongly believe that there will be a different, and those patients who got enteral feeding will be better. It's so logical. I remember that at the time we used proton pump inhibitors, nitrate patch, everything in pancreas, which later on found out that it's logical but not true. And this was the setup. One group receives 2 kilocalorie per kilogram per day, the other 0. And I have to show you it was logical, but didn't work. So this is, if you look at these two data, you can see that the endpoint was severity and mortality. Here is the severity mortality, exactly the same. So this is very important, because what I found in my journey, plenty of times basic scientists or translational scientists undervalue clinical science. They say it's not science, you're just grouping patients, and so on and so on. And exactly the same, sometimes clinical scientists don't appreciate the discoveries very much and translational. But without both of them in a high level, you can't go further. However, as I showed CFTR, we went back. And we went back again. So this is not a one life road that you start discovery going, and just one way. You need to step back, and you need to understand what didn't work. And we did another experiment, which we looked at again. And again, thank you, Marty, for the green fluorescence protein you provided us for these experiments. We looked at the CFTR, what's happening if you give ethanol. And besides ethanol, you give activator and potentiator for CFTR potentiator to the cells. And besides the ethanol, the CFTR came back again if you look at the combined therapy, and this was actually a dose dependent. And when you looked at the CFTR channel, it was a control, ethanol, and when we used the CFTR activator, the channel just opened up. So this was a very good model to show that really if you use in pancreatitis, in pancreatic models, a CFTR activator potential, it could be helpful. And we tested it in a guinea pig model that we induced pancreatitis for this and used Orkambi, because currently widely used in CF patients to activate, reactivate actually CFTR. And we were happy. This was just, it was not only published in the Journal of Physiology, but it became to be the editor's choice. And I received this letter a couple of weeks ago. However, and this is another ethical issue, I was very happy in Szeged, Ole just mentioned last week the ethical concerns. This should be also for pharmaceutical companies as well. Because, they set up the drug for one patient, $300,000, but they not only set the drug for that, but they are not willing to give anything for pancreatology. They don't want it to test, so we still need to wait until we can test the Orkambi for that. But I am sure that at one stage we will be able to test, and if yes, these discoveries, translational science will lead the very first specific therapy in pancreatitis. So this is like this, we need science and we need medicine for that. And this is my third confession. I really love Romania. And I really love being here. And when I put this presentation together, I was really surprised and shocked that already 26 brilliant young fellows came to our labs, to Szeged and to Budapest, and they came back. And now currently I am very happy to see Stefania Bunduk in the first row. She was one of the very first students who come over, and now she is a leading scientist in the Bucharest University, and they learned a lot. And now in Iași, we have, of course, friends from Iași, and in 2022, students from Iași also started coming, and currently also in Budapest in our lab, Darius, Valentin, Sandu doing research. Madalina is there, Tudor is there. Last year, Joanna were there. Joanna Irina is there. We just got the good news from one of the journals that the paper is coming for major revision. Thank you for her dedicated work. Madalina and also Diana. So they are the Iași students we are working with. Some of them are here. We continue the second, third year project. So I'm really very thankful, and I really love working with Romania. So I would like to thank you very much for your attention. Happy anniversary, and I really hope and I am sure that there will be a future of this very successful collaboration. Thank you.
Thank you so much. Ladies and gentlemen, we will have a little break right now, 15 minutes. You'll have coffee, maybe some water, yes?
So I saw that the rector is here, so just let us, the invited speakers, also to thank the Iași University, not only for welcoming us but also celebrating science, I think it. So I would like to ask Martin, Ole, András and Zoltán to come over, and please, Viorel, we would like to provide you our appreciation.
Congratulations, so thank you again, and we'll have a 15 minutes break. Thank you.
Internaționale, cu tradiționalismul și versatilitatea sa. Festivalul Internațional de Literatură și Traducere Iași, Festivalul Internațional de Teatru pentru publicul tânăr, seriile filmului românesc, Romanian Creative Week, sunt doar câteva dintre marile evenimente care pun Iașul pe harta turistică și culturală a Europei. Iașul a fost capitala Moldovei, a principatelor române și a regatului României și a rămas și astăzi, simbolic, capitala culturală a țării. Întâia universitate modernă din România, fondată la Iași în 1860, a încorporat și facultatea de medicină, inaugurată la 1 decembrie 1879, fiind astfel una dintre cele mai vechi din România. Universitatea de Medicină și Farmacie Grigore Tepopa are o tradiție de 145 de ani în cercetare și educație în domeniul medical și al științelor vieții. Înglobează o diversitate de culturi și mentalități datorită numărului impresionant de studenți internaționali. Aproape 3000 de tineri din peste 70 de țări vin la Iași pentru a-și îndeplini visul de a deveni profesioniști în domeniul sănătății.
Welcome to Yash. Welcome to Grigore Tepopa University of Medicine and Pharmacy of Yash. Different people, one university.
De Iași se leagă nume mari ale literaturii române, de la Eminescu și Creangă până la Sadoveanu sau Mihail Codreanu, ultimii doi înființând, alături de marele savant Grigore Tepopa, patronul spiritual al universității, celebra revistă Însemnărieșene. Ședințele revistei se țineau în sala Rockefeller a Institutului de Anatomie al UMF Iași, care găzduiește și astăzi faimosul ecorșeu al lui Brâncuș. Muzeele universității însumează adevărate pagini de istorie.
Vă aflați în Muzeul de Istoria Medicinei, face parte din Muzeul Universității de Medicină și Farmacie Grigore Tepopa. În aceste vitrine puteți vedea o expoziție temporară Grigore Tepopa cu obiecte personale, documente și tot felul de materiale care se leagă de trecutul său personal, de interacțiunea sa cu studenții, pentru că a fost foarte apropiat de studenți, chiar dacă era foarte sever, era un om foarte apropiat de studenți. Institutul de Anatomie, construit la sfârșitul secolului XIX, cu arhitectură specifică unui templu grecesc. În fața voastră se află una dintre lucrările importante din creația sculptorului Constantin Brâncuș. Foarte mulți turiști din străinătate vin special să vadă această lucrare. Ce-i interesant în acest muzeu și în spațiul acesta al sării Rockefeller se întâlneau marele, să spun, spirit ale culturii române, sub patronajul lui Grigore Tepopa și al soției sale, și au scos, vreme de câțiva ani, între 1936 și 1940, o revistă care s-a chemat Însemnărieșene, considerat fanion al culturii românești.
După 145 de ani, Universitatea de Medicină și Farmacie Grigore Tepopa are același spirit deschis pentru
Good afternoon. I am incredibly honored to be here at the semi-centenary of Palade's Nobel. Of course, unfortunately, I never met Professor Palade, but we're all inspired by his work and by his discoveries that really allowed us to do so many studies. And also, I am honored to share the podium with the Nobel laureate who discovered the fluorescent protein that we all used quite extensively, and of course to see Professor Pedersen again and Professor Hege. Thank you very much for joining us today. I'm also honored to be at the anniversary of 145 years of the university that I graduated myself, and where my father has been a professor dedicated his whole life. So I'm really very happy for many reasons to be here. So Professor Hege already described to you very much what I will talk about. And it's really the journey between discovery science, translational research, and clinical application, except I will change the organ. So we'll talk about liver as opposed to pancreas. Liver is also very interesting, I will assure you. And when I started working on liver cancer in particular, about 20 years ago, there was no treatment. So as you will see, there have been many, many developments, and we'll be very excited to see some of this becoming reality. So this is my disclosure. I don't have the fancy disclosure that Professor Hege had. So these are some studies that have been funded by companies, but most preclinical studies I will not make treatment recommendations. I will just show you how we can take science to the clinic. So liver, as you can imagine, is very different than pancreas in many, many ways, functionally as well as structurally. And liver cancer in particular is quite complex. So it's a disease which actually encompasses two diseases. It's the diseased liver that is caused by viral disease or alcohol consumption or toxins. And then there is the cancer that develops gradually in the liver on top of this underlying liver damage. And cancer is obviously a genetic disease. I'll talk about this later. We know quite well the mutations that lead to cancer. They're very complex, and you see them here. They're quite heterogeneous, with the exception of P53, which is lost in about 1/3 of the cases. All the others are very low frequency. We can't really target them, and cancer is a disease like no other. So you don't have one genetic mutation. Actually, you may have many genetic mutations in one cell, and you may have different genetic mutations that occur over time as the disease advances. So obviously, this presents with a very complex scenario where treatment is very difficult to predict in terms of outcome. Also, I will talk a lot about the tumor microenvironment, and I'll explain why in a second, but suffice it to say that tumors, as they develop, they acquire these new vessels, a matrix, which in turn helps the cancer, the malignant cell, to evade the immune system. And I'll show you in a second how that happens. So again, it's a genetic mutation that leads to cancer, or many genetic mutations that lead to cancer, but it will never form a tumor. In order for the cells to be able to actually form a tumor, they have to acquire these vessels with a process called angiogenesis, and then they have to have a matrix to grow on, and that's matrix production mostly by the mesenchymal component of the tumor that is also recruited, and then you have a lot of inflammation, which, counterintuitively actually helps the tumor evade the immune system, because you have all the bad players, the immunosuppressive cells. So all these processes are very much like during development, except they're all abnormal. So the vessels are very abnormal, which causes hypoxia, which helps the tumor and helps the tumor migrate and invade other places. And then, of course, fibrosis and dysplasia is also abnormal, which occurs due to cancer progression. And again, in the context of the liver, this is also complicated by the fact that you have underlying liver disease, which actually limits the application of treatment. So it's very difficult to even resect a tumor, irradiate a tumor, or give chemotherapy because the patient might not survive the treatment itself because of the damage in the liver. And again, we know all the players. Hepatic stellate cells are the mesenchymal component. They produce the matrix. And then you have a lot of macrophages, monocytes, that are the immunosuppressive cells. Some of the neutrophils may also fulfill this function. And then you have these vessels that help the tumor to grow, but at the same time, because they're very abnormal, the immune cells cannot get in to perform this function. So why is all of this important? Because liver cancer, in particular hepatocellular carcinoma, which is about 90% of the disease, is expected to increase by 55% over the next 2 decades, primarily driven by metabolic syndrome, but also by viral disease and alcohol consumption. And of course, that's the case in the United States, where we see about 3% increase per year in the number of cases, both in male and female patients, and less than 1 in 5 of these patients are expected to survive at 5 years, which is considered long-term survival. So obviously, this is a huge unmet need. So why is the outcome so poor? And this is my point of view. It perhaps is not, very politically correct, but I think we are trying to solve a very complicated problem that I just described to you with simple solutions. So, of course, we can use surgery or radiation or chemotherapy as the blunt force way of treating with the cancer, but we know that's not going to solve the problem. So we really need to find clever ways, especially with advanced disease, where we target all the components of the tumors. So the oncogenic drivers are very diverse, as I just told you. There are many, many oncogenes, so targeting one of them might be beneficial only transiently. We've seen that again and again in oncology. Then the microenvironment, we still don't understand its role. And I think for many people, it's regarded mostly as a morbidity, but not really as a mediator of progression or resistance. And I think we have learned that in liver, this is a very important factor. And then the fact that, the tumor itself is like a wound that doesn't heal. That's Professor Dvorak from Harvard defined that in the 80s. In the very nice paper in New England Journal, and it's really a systemic disease that requires complex personalized solutions that involve the host immunity. And we have now immunotherapy, which has really been a game changer, because right now, the treatments, especially in liver cancer, do not target directly the cancer cell. It's very interesting. You have this abnormal organ, and the treatments that we have today, they either target the vessels or the immune cells, indirectly controlling the tumor. So I think this is really a paradigm shift in oncology, because for the first time, we're not just going after the cancer cell to kill them. I wish we had a way to do that, but in fact, we're going directly by targeting the structure and the function of the vessels and then activating the immune cells. And to do that, I follow, in Professor Hege's footsteps to tell you that there's a very important road, and there's a huge divide between the basic research in the lab and the clinical application, because the goals of the activity of these groups are very different. Here, people are very preoccupied by discovering new genes, new molecules, new concepts, and getting mechanistic insights, where in the clinic, obviously, it's about the application. You have the cases that you heard about. You have to deal with that, so you have to see what is a treatment that is feasible and perhaps effective. So I think this is really the role of translational medicine. And this is something we started many years ago with the development of vascular endothelial growth factor blockers in the early 2000s, trying to understand in patients how this anti-angiogenic therapy works. And of course, I'm not going to bore you with the detail, but suffice it to say there are many ways to look at biomarkers, at soluble biomarkers, cellular biomarkers, or imaging biomarkers. And then for liver cancer in particular, we developed models in mice that actually reproduce many of the hallmarks of human disease. So we can actually induce liver damage and then grow a tumor, or induce a tumor with genetic means, and now do some sort of preclinical trials instead of just jumping to the patients and doing clinical trials, because we know 97% of those trials will fail. So much better than jumping without knowledge to this clinical development to actually understand this. And I'll give you some example of that. But first, let me describe a little bit in more detail about angiogenesis and why this abnormality matters. So I was very fortunate to do my postdoc with Professor Rakesh Jain from Harvard, who is really one of the the pioneers in showing what is the role of vasculature in tumors, and how this very abnormal vasculature, again, counterintuitively in a way, helps the tumor to progress, because it creates an acidotic and hypoxic environment where immune cells cannot function. And then in terms of treatment, the lack of oxygen affects radiation or some chemotherapies, and of course the delivery of chemotherapies. There's also a direct impact on the immune system, because this microenvironment abnormalities leads to production of chemokines and cytokines that recruit these immunosuppressive cells, such as T regulatory cells, certain macrophages, and inhibit the differentiation of dendritic cells into classical dendritic cell type 1, which are the ones that mediate immune responses. And all these abnormalities actually lead to reduced immune cell effector function, which is actually causing the evasion of the immune system. And then, of course, structurally, if you look, there's a very well-defined structure of the vessels in the liver, and I'm not going to go in, in the interest of time over it, but I'll just say that in tumors, this is completely lost. So what you see is a very abnormal vasculature, primarily from the arterial side, which is quite unique in the liver, and then with many areas of hypoxia. So again, 20 years ago, there were no treatments. Now I think we have too many treatments, but they don't work very well. Nevertheless, there are many VEGF inhibitor drugs, some antibodies, some kinase inhibitors that are less specific for VEGF, but specific for other kinases as well. And then we have many, many immune checkpoint blockers, CTLA-4, PD-1, PD-L1, either alone or in combination, used now for the last 7 years with very, very exciting responses in some of the patients. So obviously, the goal of treatment with immunotherapy is to have a durable response by creating this immune microenvironment that is now, as people like to refer to, as immunologically hot. So instead of this immunologically cold environment, which allows evasion of the tumor of the immune system, you would have an immunologically hot environment where dendritic cells present the antigen, T effector and NK cells are activated, and they can eliminate the tumor. That is the principle. However, it doesn't work very well. Unfortunately, less than 1 in 5 patients respond to immunotherapy, which is very expensive and very toxic. So people have tried to see perhaps we can combine immunotherapy with some of the other pillars of treatment that have been well established in oncology, such as surgery, radiotherapy, traditional chemotherapy, or other precision therapies. However, and this is something that is happening at a huge scale globally. There are thousands and thousands of trials. And from 2017 to 2020, after immunotherapy became one of the pillars of oncological treatments, you see this an increase almost triple over just 3 years in terms of combinations. Majority of them are with chemotherapy, but also combinations with anti-angiogenic therapy, combinations with radiation, and so on. What I feel is, going back to Professor Hage's talk, this is not really biomarker driven. We're just throwing darts and hoping that we would hit the target. I think what we really need to do is really be able to conceptually go after a specific mechanism or specific selection criteria that will allow us to hit the target more often than we do these days. So in summary, hepatocellular carcinoma are very heterogeneous, aggressive, resistant liver cancers. The therapeutic benefits depend on cancer control, but also treatment feasibility. The abnormal vessel immune suppression mediate disease progression and treatment resistance. And then increasing immunotherapy efficacy, which is quite impressive in some patients, will require combination strategies. So I want to tell you 3 very short stories. One is about anti-VEGF immune checkpoint blockade combination and why this works and why it has become the standard of care in patients. And then I want to talk in very short stories about targeting a chemokine receptor called CXCR4 and also restoring P53 function as the last story. So let's start with the VEGF blockade. So again, Professor Jain was my mentor, still a very close collaborator. He received the Medal of Science for this concept where he showed that in fact VEGF anti-angiogenic therapy doesn't work by pruning the vessels and starving the tumor, but instead by normalizing the vessel function, which was very relevant, not just for immunotherapy, but also for chemotherapy. Unfortunately, there is a normalization time window here, which really depends on the dose and the duration of treatment. So this is what he postulated about 25 years ago. This has been shown to be true, and it's one of the founding principles of these combination therapies. So we started doing that in the patients about 20 years ago with kinase inhibitors for VEGF as they were developed at that time. And what we found that indeed There was a normalization of the vessels. There was a decreased permeability, which is usually caused by VEGF. So the K-trans is a parameter you can measure by MRI. We are able to show that indeed in patients you can decrease the permeability and the surface area of the vessels, which is so abnormal in ACC. It's kind of normalized the vessels. However, this was, again, transient and didn't really translate into a benefit. And then when we did all the biomarker work with Professor Zhu at the time in patients and published this about 10 years ago, the reason for this resistance of the tumor was really by activation of some of the inflammatory pathways that were leading to immune evasion instead of immune activation. So then one of them was SDF1 alpha, which binds to CXCR4. So I'll come back to that later on. So then obviously the question was, can VEGF blockade be combined with immunotherapy to be more effective? So instead of VEGF blockade, inducing immune suppression to see if we can activate the immune system and see whether that's the case. So this is the result of about 10 years of work, so I'm just going to summarize it in one slide. But I think it's very important for what's going to follow, because what it showed is that when you combine antibodies against VEGF pathway and PD-1 pathway, actually what you have is similar to a wound that is healing. You have a normal vasculature reforming. So it's like, the lymphocytes are coming in, they're eliminating the tumor in an environment that is normalized, and now that helps the vessels form in a normal way, so the liver is trying to regenerate itself. So I think that was, very difficult to convince the oncologists that that's the case. However, they did a phase 3 trial with VEGF antibodies and PD-L1, and that showed unprecedented efficacy over what was at that time a kinase inhibitor that was used extensively, sorafenib, with more than 19 months compared to sorafenib, which was about 1 year. However, moreover, when they did a lot of correlative studies, they indeed found that it was very important that vascular normalization occurred, and some of the abnormal vessels were eliminated, and the vessel function was restored in this patient in order for the treatment to work. So that was really exciting, and now we have 7 different combinations, both antibodies against VEGF and kinase inhibitors, that are approved across different indications. I'm sure that this number will increase further in the years to come. However, for kinase inhibitor in particular, in the context of liver cancer, we've been faced with the problem that a lot of the clinical trials actually failed. And nobody really knew why this failed. And I think, to us, one of the main reasons is that these drugs are not really specific to VEGF pathway. They target many other pathways that tend to destabilize the vessels instead of normalizing them. So we looked at the impact of changing the dose or changing the scheduling of these kinase inhibitors to see whether we could improve that and whether that's going to lead to better outcomes. So in fact, about 10 years ago, we published this paper that showed very clearly that with the use of kinase inhibitors such as sorafenib, which inhibits PDGFR and many other factors that are important for vascular stability, one can actually aggravate hypoxia, causing immune suppression, and this was actually prevented by blocking the CXCR4 pathway, again, very central to immune suppression. And only this way, this worked with PD-1. So this was very basic work. It wasn't really easy to translate clinically, but it showed the importance of reprogramming the microenvironment and preventing this vascular dysfunction. So to achieve vascular normalization, what we did is actually we looked for the dose, the optimal dose of the kinase inhibitor that would stabilize the vessels, have more mature vessels as opposed to pruning the vessels, and we found that there was indeed an intermediate dose of a kinase inhibitor that achieved that in mice. And then with this intermediate dose, you could actually see a very large infiltration by effector T cells. This wasn't seen with all the other doses of the kinase inhibitor. This was, at the time, this was also viewed as some sort of mouse experiment, and it was very difficult for oncologists to see how this would actually be used clinically. However, before I get to the clinical application, that also increased survival, and we showed using knockout mice and different PK, PD studies, or the mechanism. I'm not going to go into the detail, but suffice it to say it had to do with vascular normalization and accumulation of the drug of the immune cells in the tumor. More importantly, when they did the clinical trial, actually with a lower dose, intermediate dose in Korea, there was a phase 2 study. This was published earlier this year in Nature Medicine, showing that indeed 1 in 3 patients had a complete or partial response, and overall survival in this very advanced disease patient was more than 80%. So very exciting data. This is now, excuse me, being tested in a phase 3 trial. Then we also asked the question if this immunotherapy and infiltration of lymphocytes can really stabilize the vessels, can we perhaps start with immunotherapy and then give the VEGF as opposed to what historically has been done just because of the way they were discovered. First using VEGF and then PD-1. We asked the question, can we reverse that and give the immunotherapy first to prepare to prime the tumor and then give the kinase inhibitor. And indeed, this led to more than doubling of survival in mouse models using 2 different kinase inhibitors. This was also published a couple of years ago and now is being tested in the clinic with our collaborators in Hong Kong. So lastly, for VEGF, I want to talk about another very exciting concept. Can we bring this intervention earlier? So again, this is nowadays used for advanced disease. The probability of achieving cure or even extensive survival at this stage is very low. So then the question becomes, can we bring them in earlier stage disease to increase the chance of curing the patients and then also increase their survival? Of course, that also means that we need to understand a little bit better the difference between early disease and advanced disease and whether the same mechanisms that I described are relevant there. We don't really know. But some of the work that we've done with our collaborators in Bucharest, with Professor Popescu in particular, we found that indeed angiogenesis and inflammatory pathways are very relevant even in the patients with early disease that are candidates for surgery. This study wasn't really looking at that. They were just trying to find how do we stratify patients for resection or transplant. However, what we found accidentally here is that angiogenesis and immunosuppression are very relevant early on. So we are doing now a randomized phase 2 study where the patients receive VEGF and PD-L1 blockers before surgery, and then they go to resection. Again, this is early disease, resectable. However, some patients go directly to surgery. Some patients have these interventions for 6 cycles before surgery, which not only, hopefully, will increase the curability and also the survival of these patients, but it will allow us an incredible opportunity to do a lot of tissue studies, a lot of biomarker studies, a lot of image studies to really test whether all these hypotheses that we put forward in the lab are correct. So we're very excited that we're about halfway through the study and hopefully we'll be able to look at that soon. And also, the other important thing is, again, the difference between early disease and recurrent disease. So with our collaborators in Guangzhou in China, actually, we had the chance in surgical patients to look at the environment of patients who recur by intrahepatic metastases versus those who had de novo recurrent HCC, meaning a cirrhotic nodule became malignant. So very interesting. Of course, the genetics were different because they were different tumors, but in fact, the largest difference was in the microenvironment, and it had to do actually with PD-1. So in the de novo recurrent HCC, PD-1 was expressed, TIM3 was expressed, the T cells were exhausted. However, in the truly metastatic disease, there was no PD-1. The T cells were not exhausted. They were just not anergic, and there were no dendritic cells where they were not mature at all. So obviously, very different scenarios will require different treatment. However, all these patients are treated the same these days, for example, with PD-1 inhibitors. So Professor Kwang, who is the PI there, he did this phase 1 study where he characterized the tumors first and then gave a PD-1 inhibitor. As we predicted, only the patients with de novo ACC responded to PD-1 blockade and no those with the metastatic disease. So now he's obviously stratifying the patients and is doing a phase 2 to validate these studies. But that also raises the question, how do we predict and monitor immunotherapy responses? So one of the ways is using fluorescence. We heard from the pioneer of the use of GFP. Unfortunately, I apologize, but this is a red fluorescent protein. So this basically is labeling PD-L1 antibodies, which allows you now in vivo to monitor PD-L1 expression, because PD-L1 expression is inducible. So it's very hard to have a tissue section and be able to give a diagnosis. This is showing dynamically PD-L1 expression also allows you to localize where the PD-L1 is expressed. So I think hopefully this will also help stratification. We hope to transfer this to the clinic. This was a study in mice. Another very interesting finding that has created quite a lot of interest lately is this the tertiary lymphoid structures that occur in tumors. So it turns out that tumors have their own lymph nodes where T cells and B cells coexist, and they may be actually mediators either of treatment response or treatment resistance. So I'm not going to go over the details, but this is one of the very interesting research direction may provide you a biomarker if you have access to the tissue, and perhaps if we can find new imaging ways of looking at TLSs, whether the HCC will regress or not after immunotherapy. How about the dendritic cells? I repeated several times that you have very few mature dendritic cells. Is there any way to now activate these dendritic cells? So we were, again, that was somewhat serendipitous finding that we were following up on this CXCR4 blockade with antibodies. I apologize about the, this is a Mac on a PC, but we were doing this blockade of CXCR4 with PD-1 in combination, and we found some unusual synergy with a lot of tumors responding even after treatment was stopped, with a lot of prolonged survival and with a very aggressive HCC model. And then when we did a lot of the transcriptional analysis, I don't have time to go over them, but I will just tell you that some of the top genes that were increased had to do with the maturation of dendritic cells. So clearly with this combination, we were able to actually mature the dendritic cells, also bring them in the tumor. So that was the case when we started using immunofluorescence to look for these. We found that indeed both in grafted as well as in spontaneous HCCs, you see a lot of dendritic cells that are mature and CDC1 type, and this was significant both in the grafted and in the spontaneous tumors. And when we did this study in mice that lack genetically this population, which is called BATF3, knockout mice, this was one of the genes that came out from the transcriptional analysis, the whole benefit was gone. So very surprisingly to us, in fact, CXCR4 benefit was entirely because of the maturation and activity of the CDC1's dendritic cells in this context. And in fact, we did a lot of in vitro studies to show that indeed both PD-1 blockade and CXCR4 were very important for dendritic cell maturation. And this is one of the ways that perhaps now we can mitigate this issue that dendritic cells are not there to present the antigen to the T cells. So the last story that I want to tell you over the last minute or so is about P53. As I said, this is probably the most common genetic mutation. It's a loss of function in 36% of the cases, not druggable, considered for decades. This is something that we developed with our colleagues at Brigham and Women's Hospital, also from Harvard. Professor Shi developed these nanoparticles that can target the hepatocellular carcinoma and restore P53 function with mRNA. So it's a transient restoration of P53 expression. We showed that indeed that could induce apoptosis. You can combine it with some other pathway inhibitors. But then we started looking at immunotherapy as well, because immunotherapy became so relevant for ACC. And this was also published a couple of years ago. So let me give you the details very briefly of that. We used this P53 restoration that on top of that would block PD-1. You can see here that P53 was now expressed in his ACC that didn't really have any P53 expression. And that actually led to quite substantial inhibition of growth when you combine P53 with anti-PD-1 immunotherapy. And then we did a survival study to see whether that's going to really increase the survival, and that was the case. Each therapy alone worked a little bit, but then there was a very substantial increase in survival with the combination, and that was because not only we inhibited tumor growth, but it reduces a lot of morbidity, such as ascites, pleural effusions, and a lot of the metastatic disease was reduced. So with that, we did a lot of analysis to see what happened to the immune environment. And again, in the interest of time, I'm not going to go over the details, but suffice it to say that the tumor became more visible because MHC class I was increased on the cancer cells, and there were a lot of changes that are associated with the immune response, such as activation of CD4 and CD8 cells, and increase in number, and also a more favorable ratio of different types of macrophages. So with that, we're taking this concept to the clinic now with the DoD impact of the Department of Defense award from the United States, where we're using these nanoparticles that are used clinically, liposomal particles, to restore p53 function and combine it with what is now standard immunotherapy for ACC. So we're very excited about this development, and hopefully it will also show a way of how you can combine targeting the cancer with immunotherapy. So with that, I'd like to conclude and say that normalization of the immune environment is critical for durability of responses of systemic therapies. We know that to be the case now. I think immunotherapy is very, very exciting, has really changed the landscape in oncology. However, we really need to be more careful about how we select patients, how we understand, based on the understanding of the treatment interaction, which has to be based on biology, and hopefully this will lead to more effective and safer therapies. So with that, I would like to thank, first of all, the organizers for allowing me to speak here, and then many people who contributed to this work, both in my lab and also wonderful collaborators across the world, including here in Romania, and of course the generous funding we received, and thank you for your attention.
Thank you so much. We will continue, ladies and gentlemen, with Professor Dr. Cristiana Petrei. He is a professor of infectious diseases and microbiology at the School of Medicine, University of Pittsburgh, United States of America, and we're glad to welcome him here at UMF Iași.
I need to start with apologizing for not being here earlier, but my life is very complicated now. Also need to do the disclosures. We've only been funded by the NIH, and with the arrival of Robert F. Kennedy, probably there will be no disclosure soon. And finally, I would like to apologize for not talking about science, but about the lack of science. And the title is French because it rhymes. In English, it's The History of Madness and Times of Pandemic, a very appropriate title for our days. The title is obviously inspired by Michel Foucault's History of Madness, in which Michel Foucault says people know what they do, frequently they know why they do that they do, but what they don't know is what they do does. Very easy to understand, but it's very inspiring too, and translated in Romanian, and I apologize for all the guests, it translates very well in a word which is called zorbalık, which is kind of social madness, which occurs periodically associated with pandemics. The origin of this talk was generated by this quote from a very important book. I gave it for the last 4 years, probably in most of my talks, which stated that as the pandemic evolved, you could... it's here? No. Oh, no, so this is... Sorry. But I don't have... Oh, okay. Oh, sorry. During the pandemic, we saw people gradually turning away from conventional medicine. That pandemic showed how important is the trust between people and doctors and with the public health experts. And also the pandemic illustrated how absolutely critical to so many aspects of healthcare is trust of general public, medics, and their governments. You resonate with this, but this is not related to the COVID pandemic. This is related to the Spanish flu pandemic. So suggesting that we don't learn so much from history. And actually I'll show you now a lot of examples that we don't learn anything from history. So we've been just confronted with one of the major pandemics of the last few centuries, in which we did good. We developed in record time diagnostic tests, therapeutics regimen, and preventative measures. Virus characterization was completed in 1 month, so we had the sequence of the virus before most of the people knew that we have a pandemic. We generated 18 animal models within 6 months since the discovery of the virus. Up to now, we generated more than 16 million viral copies. I have a friend which works in Glasgow and is in charge of the SARS-CoV-2 sequences, and he's saying, for the last 4 years, for me, it was Christmas. The first million HIV sequence, which was the second most sequenced virus in the history, was completed in 40 years, just to have an idea. More than 400,000 published papers. To an unprecedented convention, we got full access to all these papers through publishing houses. Bill Gates, of course. We established much more than before, the preprint use of preprint service, which made the information available immediately after the paper was ready. We generated 4 vaccines, which were available in less than 1 year. A lot of monoclonal antibodies, antiviral, everything was available in a very short period of time. So we did great. In the meantime, the pandemic triggered a parallel epidemic of pseudo-information, disinformation, manipulation, and in general, unprecedented social anarchy. Anti-vaccine movements reach unprecedented popularity to a point in which they can endanger now the national vaccine programs, and we're talking not about SARS-CoV-2, we're talking about measles, we're talking about polio, we're talking about a very important vaccine programs. There were a lot of need for freedom derived prevention policies, especially in some countries that we know, United States, south of the United States, Romania, and others. Aberrant and ridiculous and embarrassing judiciary complaint effectively prevented implementation of preventative measures. For instance, the AD of Bergamo decided that the doctors didn't treat patients well in Lombardia. Good. It was a very, very gifted AD. Not even the diagnostic tests. Supreme Court of Lisbon judged real-time PCR as being ineffective for diagnosing SARS-CoV-2. Now, for the people which are not Romanian in this room, I have to tell you the Court of Lisbon decided that the bastard of the Romanian throne was the legal successor. So we know very well Court of Lisbon. Social media promoted treatments without solid scientific validation, generated confusion, and impacted the vaccination programs. This is really bad. To a point in which WHO generated this word infodemic, the portmanteau derivation from information and epidemic, which is defined in the WHO document referring to a rapid and far-reaching spread of information about epidemic, which may be accurate, inaccurate, misinformation, deliberately misleading disinformation. As fact, rumors, and fears mix and disperse, discerning between the real and inaccurate information becomes very difficult. I mean, and this, we really saw it. I mean, a lot of people that decided that they will do good things, were under so much pressure that they started doing very bad. So, to all these questions, we have to state this. Those who can't remember the past are condemned to repeat it, and we repeat it with every pandemic in the history of mankind. And I'll show you some examples which are pretty suggestive. Throughout our history, pandemics changed the course of mankind history. Oh, sorry, again. During the Middle Ages, succession in the lines of throne were repeatedly altered by epidemics, mostly plague and smallpox. The extension of civilization, so the Columbian exchange, was supported by disease exportation. To resume this in a single phrase, Europeans gave Americans smallpox, Americans gave Europeans syphilis. The First World War was lost because the Spanish flu, the German general, which was in charge of the Meuse battle, lists this in his memoir. They lost the battle which they were supposed to win easily because the vast majority of the German troops were sick. Okay, and now I will go with the major characteristics of the pandemics. The history of pandemics is a history of changes. Very interesting. In a time when a lot of climate deniers are taking a lot of traction, climate change are associated with pandemics. The first plague, the first ever pandemic or epidemic, the Chinese epidemic, occurred in a period that coincided with the Holocene megathermal, a period of warmth that lasted about 4,000 years until 3,000 years before Christ, and which was followed by a gradual increase in temperature of about half degree per millennia. Now we are increasing about 1 degree per century. What we call the Kokolitslo outbreaks, actually this is the outbreak that decimated the American nations. First smallpox, but then two successive epidemics which are associated with severe drought decimated the natural inhabitants of Hispaniola. Actually, the population of Hispaniola, which is actually the island represented now by the Dominican Republic and Haiti, was reduced from 20 million to 100,000 in only 100 years. By diseases mostly, not by the conquistadors. The history of epidemics is a history of political changes, which we name now in all the conspiration theories great resets. Yeah? There were a lot of great resets in history. The golden age of Athens ended with the Athenian plagues. After the plague, Sparta took power and Athens never took it back. Pax Romana, a 200-year period of peace, was ended by the Antonine plague, and I'll detail all this later. The Justinian plague contributed to the end of antiquity in general. There were little resets, dynastic changes. I'm listing here just a small part of them. My favorite of these ones is this. Prince Albert Victor, Queen Victoria grandson, second in line of throne, died of Russian flu, a disease which main symptoms were loss of smell and taste. Okay. There were also great medical recesses, great medical progresses that were always associated with pandemic. Middle Age plagues gave birth to the quarantine. The Urine plague contributed to the birth of preventative medicine, and I'll come back to this. Spanish flu gave us Lysol. Typhus pandemic in Warsaw Ghetto demonstrated how crucial for successfully controlling an epidemic is social cohesion. And SARS-CoV-2 precipitated the implementation of mRNA vaccines. So we really made a lot of progress under the pressure of pandemics. Now I'll show you some examples of, the last one is the political unrest. During the Athenian plague, the measures were not implemented properly because some of the inhabitants lost their moral principles. Such as now people wanted very much to go to do snorkeling in Belize instead of staying in controlling the diseases. During the Antonine plague, the shortage of gladiators generated political unrest in Rome because people had no bread and they wanted a lot of circus. The history of pandemics is a history of disinformation. This always functioned very well, spreading rumors, false remedies, false sources, missteps of the doctors really generated the unrest because people perceived those missteps of the doctors, which were learning the disease as the people were learning it, as things that the doctors hide from them. In the entire history of mankind, this feature characterized all the pandemics, but never before at this level that we experienced it now when we have social media. The examples, the heated plague is very important. The first use of biological weapons. So during the Hittite-Arzawani War, the Hittites sent into the Arzawan camp a herd of sheep which are infected with Francisella tularensis. And they won the war because of the tularemia, not because of their war success. Francisella was actually endemic at that time, and we know it, and you know it, you saw it, and I'll show you where you saw it. The Philistine disease, yeah? The Philistine disease was contained in the Ark of Covenant. So, which means that what you saw here killing the Nazi in Indiana Jones was tularemia, very acute disease. Yeah. The Athenian plague suddenly ended the Pericles century. That was the golden age of Athens. The great artists, Phidias and Myron, great writers, Aeschylus, Sophocles, Euripides, the great philosophers, Socrates and Plato, and even the greatest doctor of all the time, Hippocrates, of course. These guys were contributing to the golden age of antiquity. And the Athenian diseases contributed to end it. It lasted 5 years, killed about between 75,000 and 100,000 people, 1/4 of the population of Athens. What was really important was that during this time, the Athenians were, again, not working. The Athenians were at war with Sparta, and after the disease, they lost the war. The Athenian plague was very difficult to control because a fraction of Athenians did not observe the restriction responses. I mentioned the Thucydides saying, I mean, now there are a lot of people, political parties that are discussing, but this is the greatest, one of the greatest historians of antiquity, saying that people who don't follow the restrictions imposed for controlling a pandemic lost their moral principles. So the alterations of the social fiber and the instability and the lack of empathy, which is also what Pericles told to the Athenians, contributed substantially to the loss of war. The next one also had a very prominent doctor to control it, was the Antonian plague. Entered the empire, don't know exactly, somewhere from the eastern part. There are a lot of myths on the origin of the plagues. The Romans did a lot of bad things and released the disease. Even the Christians were infuriating the gods because they don't observe them. But anyway, the good thing, the only myth that we have is that Bill Gates generated this plague. But the fact and the matter is that between 2 and 33% of the population of the Roman Empire died, which is a lot of millions. Mortality rate was about 10 to 15% in the cities and among the soldiers. Probably, we don't know, probably a smallpox. The symptomatology is also suggesting for the smallpox, but the lethality is slightly lower than what we saw with the smallpox. The fact and the matter is that 1/4 of the Roman Empire population died. The doctor, the Fauci of the Roman Empire, Galenus gave us a very, very good description of the disease. What happens is that at the time when this epidemic hit the empire was also attacked from the north by the Germans. Germanic tribes, sorry, not the Germans, the Germanic tribes. And Marcus Aurelius, very important emperor, decided to recruit slaves, gladiators, and people from the prisons. They all were promised freedom just to fight on the northern frontier. They fought very well. They won the war not necessarily because they were so effective, but because the Germanic tribes were also hit by the disease. But anyway, a few years later, the Germanic tribes ended the Western Roman Empire. The Justinianic plague is probably the most dramatic and is the first one for which we know the etiological agent. It was Yersinia pestis. It's confirmed, isolated from cemeteries at that time. Also had a very important historian, Procopius of Caesarea, that gives us a lot of description, with an absolutely clinical description of plague. The disease decimated between 20 and 40% of the population of the Roman Empire. What's really interesting with this disease is that this is the first time when we see denialists coming in. So actually, when COVID hit, there was a paper published in PNAS which called Justinianic plague an inconsequential pandemic and rejected the catastrophe, saying it was a simple cold. No more severe than a flu pandemic, mortality levels were absolutely severely exaggerated, and the pandemic was inconsequential. so important this paper was that the Guardian wanted to redo the picture of the Roman guards in the front of the Colosseum, saying that the Romans were not killed by this pandemic. Unfortunately, the Guardians, as the historians that said this, dressed the people in the uniforms of the first century Roman guards. This was the level of their knowledge. There was a huge debate between historians regarding this pandemic, very interesting features. So the argument of the historians were there were no law texts related to the pandemic, but actually the others observed that during the first half of the Justinian reign, Justinian passed 530 laws, while in the second half he only passed 19 laws, so 35 per year versus 1 per year. There was a reason for not passing the law. The second argument was among the 4,000 papyruses that could be located and identified, only 26 refers to the plague, so it's not important. The counter-argument was that most of these papyruses are actually expenditure lists, so there was no much. And probably the most important one was that the plague didn't pass Constantinople. You have here places from where we have sequences, Yersinia sequences all over the Europe. Only one in one, only one sequence in one tomb in Wales. And this was actually the most important argument that Justinianic plague was not important. You go in a very important mass tomb and you find only one sequence of Yersinia pestis. And I'll give you literally, textually, the response of a very, very great specialist of Justinianic plague, because it's really important to see. So Peter Cyrus, certainly, if one were to find the body of an extraterrestrial buried in the Welsh hillside, one would hardly dismiss it with the words along the lines, well, it's only one alien. One would want to know how it got there, and one would have to work on the assumption that the Voyager probably had not traveled alone. Yeah, so yeah, there was an important pandemic, and every single document demonstrates that the Justinianic plague actually ended the antiquity. For the next 1,000 years, plague was the major pandemic and came back in Europe in several major instances. Probably the most interesting of all is the plague in Marseille, because we learned a lot of things. So Marseille was experienced an outbreak of plague about 50 years before the Great Plague of Marseille, and they established very well-defined and very detailed measure of quarantine. Levels of quarantine, where the ship which went through a harbor in which there were cases but didn't stop, where the next ship which got to stop in a harbor with plague, where a ship with patients or casualties on board should have gone to be quarantined. Everything was respected until the great medieval affair of Beaucaire was supposed to occur, and the merchandise was not there. So all the businessmen from Marseille went to the mayor and said, okay, you have to leave this ship, where is Grand Saint-Antoine, to enter the harbor. The ship passed Smyrna, Tripoli, and Cyprus, all three experiencing heavy plague. A Turkish passenger died on the ship together with several crew members, including the surgeon. The ship was denied entrance in Livorno because it was so dangerous. It was initially quarantined and then released. In 2 years, about 100,000 people died. What was really very interesting and important in this story is, where is it? No, I didn't. I took it out. There was a lockdown 50 kilometers north of Marseille at Toulon, where the entire French float was located, and they didn't lose their fleet too. The quarantine was born in Dubrovnik. The first isolation was observed in Ragusa, but it wasn't a real quarantine because it was only 30 days, so it was a Trentin, and it was not generalized. So you could get into the quarantine, but if you are paying, you are released on the land, and it didn't work so well. But then the 40 days was established based on Christian religious significances, and it was completely applied to everybody. And the incidence of plague significantly decreased in every in every harbor. Very important is the story of the peasants in Aram. Aram is a village which is located on one of the most important trade roads in Great Britain. One case occurred with blankets brought from London. In 6 weeks, 29 people died. This is where Aram is located, so you see it's between Birmingham and Leeds. The priest, the vicar of Aram, William Mompesson, decided that it's the time that they enter in quarantine. He was a Puritan. They didn't like it so much in the village because he replaced the previous one. But then the two vicars addressed together the city, and they established the voluntary quarantine. 14 months, nobody entered and left the village. They established a sanitary perimeter. So they invented the preventative medicine. They established a sanitary perimeter 1 mile outside the village. People were not, people from the vicinities never passed the perimeter, but they were leaving them food and money because they were sacrificing themselves. 260 out of the 800 inhabitants of the city died, and it was really bad. What they did, they established social distances. Masses were held outside. The corpses were not transported on the street. They were buried directly without any necropsies, as the people claimed here during the COVID pandemic. And a lot of them started sleeping outside of the village. One of their descendants became a member of the Chamber of Lords and compared their sacrifice to the sacrifice of the heroes of Thermopylae and Marathon. The Russian plague is also one of my favorite. Not very many casualties, only 1/6 and 1/3 of the population. The problem is that it was started at Iași, here. And probably not very many people know that Potemkin is buried here in Goliath's church. It was great. Russians feel so much need to take care of their people which are dying of plague that they inherited from here that they took Bessarabia with them in order to take care. And Bessarabia still is with them. Anyway, very interesting things. They established lockdown, which is really an irony of sort. All the Russian propaganda during the COVID pandemic was the source of every movement against the lockdown. Lockdown was invented in Russia. The military were having very clear protocols for diagnostic and treatment, the protocols. They had a lot of they established the training for this. How did they get funded? Any idea? Through the taxation of vodka. Okay, next. How did the Westerners fought the plague? This is the Easterners were doing lockdown. The Westerners fought the plague through the work of this guy. This is the only paper he ever published in his life. The natural history of cuckoo. I mean, the real cuckoo. When he tried to publish his second paper, this is the reviews he got. The Royal Society of London is keeping this record. The paper was rejected because it was a deviation from known data. It was incredible, and the author should not promote such ideas because they will affect his reputation. The reputation given by the cuckoo paper. The paper that was rejected was this one. The paper in which Jenner was describing vaccination, the birth of immunology, the birth of vaccinology, the birth of every control that you can talk. But these are the two reviewers, reviewer 1 and reviewer 2. History, which means that every time when you want to kill a reviewer, you have to think of the history of Jenner. Are the vaccines important? Are very important. This is a study that my colleagues of the University of Pittsburgh published. You can see here the heat maps, how the vaccination actually cleaned the major diseases and how many cases were prevented. Altogether, by studying only the recorded data in the United States, between 76 and 106 million of deaths were prevented in 120 years. Now, the problem is that, as you see here in this figure, we are exposed to over and over and over to new diseases, and we will need to take care of those. So the last question I would like to address in the last few minutes, why are the Westerners so resistant to vaccine? Because it's obviously, it's one of the easiest medical interventions, the most successful in the history of science, and yet we have a lot of people which don't want to be vaccinated. First, you have to understand that Westerners have an intrinsic resistance to new discoveries. Some of the examples, when coffee was introduced in Europe, monks were saying you should not drink it because it will make you sterile. For the people which are not Romanian, you have to know that the COVID vaccine can make you not even sterile. It will make you sterile for 3 generations, which is a very important concept. Steam engines were reported that to potentially slip off the rails. Electric bulbs were burned and generate fires in the cities. This is the time when cities were illuminated by gas, and they were burning like, I don't know like what, but they were burning like London, like San Francisco, like everywhere. GMOs are very bad for human health. There is no proof for this. But we know that GMOs are more resistant to parasites, therefore they need less pesticides, which means that they are less cancer prone, and cancer we know, I mean pesticides we know are carcinogenic. There was a fierce resistance to the use of the most revolutionary vaccine technology during a pandemic that killed 7 million people. Anyway, there is a general resistance to vaccination. And there is this amazing book by Callistus Juma, which established why people are resistant to innovation, and I wanted to list it because it's really important. Humans oppose innovation even in situations in which this is against their own interest. Technologies that are clearly superior than the previous one are easily adopted. They tend to adopt easier technologies that are they give them more autonomy. This is why everybody is complaining, I mean all the conspiracionists are complaining about the vaccine that may use the 5G technology while they are using phones which are absolutely using the 5G technology. The sources of the resistance are multiple and they are listed here. People adopt decisions related to the new innovation are based on gut feeling rather than other evidence. People do not fear technologies but their personal losses. Scientists never consider the impact of their discoveries. So for instance, now scientists are hurrying to develop AI technology, not considering that AI technology will lose us 1 billion jobs in the next years. And very important, politicians always believe that innovation is linear, while the innovation is exponential. And therefore, the governments do not have programs for requalification of the workforce to assimilate innovation. So to conclude, originally I wanted to have a very cultural quote from Gabriel Garcia Marquez, but you have to put quotes in the context, so probably the most appropriate quote for this is, in a word which is turned down, is this one. Debating an idiot is like playing chess with a pigeon. It knocks the pieces over, craps on the board, and flies back to his flock to claim victory. Thank you very much.
Thank you very much as well. Ladies and gentlemen, as we come to the end of this special celebration, let us take a moment to reflect on the remarkable journey of the University of Medicine and Pharmacy from Iași over the past 145 years. Today we have honored our past, celebrated our present, and looked forward to a future filled with continued excellence. We are grateful to all who have contributed to the legacy of this great institution. Thank you for being part of this unforgettable event, and may UMF Iași continue to shine as a beacon of knowledge and innovation for many years to come. I'm Laura Lucescu, and I'm delighted that I could join you in this celebration today. Have a nice evening. Goodbye.